Morning, everyone. Thank you for joining us today. I think we have a good number, so we're going to go ahead and kick things off. My name is Megan Miller. I'm Director of Outreach for Critical Path's Rare Neurodegenerative Disease Program. We are thrilled to welcome you all to today's research update meeting, which brings together partners from across the ALS and broader rare neurodegenerative diseases research community to share progress on a range of collaborative efforts. This includes, but is not limited to, work supported through the public-private partnerships that were established under the Accelerating Access to Critical Therapies for ALS Act, or Act for ALS. Tanya, if we wouldn't mind advancing the slide to the agenda. Oh, we have to do housekeeping first, sorry.
No worries. No, just thank you, Megan. Just a couple of reminders and a couple of housekeeping tips before we get started with today's discussion. One, if you have any questions for our speakers and panelists throughout their presentation, please make sure that you put those into the Q&A chat box. All of our participants' lines are muted to prevent background noise. This is just a reminder that our presentation is being recorded and will be made available on CPAT's YouTube channel. And then finally, if you would like additional information or if there are questions following today's webinar, please feel free to email our CPR&D functional email box, and we will send that information out as well as a reminder. But Megan, back to you for welcome and an overview of our agenda.
Thank you, Tanya. So our team shared a detailed agenda in advance of the call, so I won't go through it in too much detail this morning, but I do just want to take a moment to highlight that the work you'll hear about today reflects collaborative efforts across Critical Path Institute, the U.S. Food and Drug Administration, the National Institutes of Health, the Foundation for NIH, as well as numerous patient and advocacy organizations and members of the lived experience community. We collectively recognize that bringing these complementary perspectives and areas of expertise together is really essential to advancing progress toward our common goal of accelerating research and therapeutic development for rare neurodegenerative diseases. We are also pleased to welcome a large and diverse group of attendees to the webinar. We had, as of last night, over 280 individuals register to attend today, including researchers, funders, industry partners, regulators, advocacy organizations, and members of the lived experience community. So thank you again for being here. We are really looking forward to a productive and engaging discussion. To kick us off this morning, it is now my absolute pleasure to introduce our opening speaker, Dr. Teresa Baracchio. Teresa leads the Office of Neuroscience within the FDA's Center for Drug Evaluation and Research, and she has been a long-standing leader in advancing regulatory approaches for neurodegenerative diseases. We are grateful for her leadership and partnership in this work. Thank you, Teresa, for joining us, and I will kick it over to you.
Thank you very much for that introduction, and good morning and welcome to everyone online. And as you heard, my name is Teresa Baracchio, and I'm the director of the Office of Neuroscience in the Center for Drug Evaluation and Research, which is also known as CDER at FDA. And I'm very excited to hear that we have so many attendees online today. And on behalf of all the partners who are working together under Act for ALS, I want to thank you all for all the enthusiasm and support you've provided for Act for ALS to make it a success. So looking back, on December 23rd, 2021, the Accelerating Access to Critical Therapies Act for Critical Therapies for ALS Act, which is also known as Act for ALS, was signed into law. And as a refresher, the law was intended to accelerate the development of therapies for ALS and other rare neurodegenerative diseases by bringing together FDA, NIH, and other partners, and by providing funding for activities under the Act. The core elements of the Act were the establishment of a public-private partnership for the purpose of advancing the understanding of neurodegenerative diseases and fostering the development of treatments for ALS and other rare neurodegenerative diseases. Grants to support expanded access programs of investigational therapies for ALS, an FDA rare neurodegenerative disease grant program, and then a 5-year action plan from FDA for regulatory science initiatives that would foster the development of safe and effective drugs for ALS and other rare neurodegenerative diseases. So all of these core elements, we've successfully achieved meeting those elements, and we have, through that, established a strong framework for building additional initiatives in the future. Under Act for ALS, GAO was directed to develop a report approximately 4 years after the Act was enacted to report on how the funding was spent and the impact of that funding. And that report was released earlier this year in February. I suspect that many attendees on the call today have probably read that report, and the meeting today will help bring some color to that report and just give some live feedback on what those activities described in the report are. You're going to hear today from different partners who are leading these efforts, the accomplishments to date, what efforts are still ongoing, and identify opportunities for future work. There will be opportunities during the meeting today to ask questions about the work that has been done. And at this point, I'd like to acknowledge the many collaborators on this effort who have been integral to the success of Act for ALS. So, of course, I'm representing the FDA, and I'm a member of CEDAR. However, at FDA, this has been a cross-center and cross-agency effort with representatives and members of our group from across the different medical product centers in CBER, our biologics group, and CDRH, our devices group, as well as the Office of Orphan Products. NIH has been the other foundational partner here, and in particular, NINDS has been leading the efforts for NIH. The Foundation for NIH has helped support the AMP ALS aspect of the public-private partnership, and the Critical Path Institute, who's hosting the conference today, has been another partner under the public-private partnership with the Critical Path Rare Neurodegenerative Disease Consortium. And then there's also the All ALS Consortium. And of course, critical members to all of this have been the many representatives from the patient and advocacy community, as well as academia and industry who have been participants in our public-private partnerships. So today's presenters are very excited to share with you all the great work that we've accomplished under Act for ALS. I think everyone on the call, on the webinar today, will see the shared energy, commitment, and sense of urgency that our partners bring to the activities that have been conducted under Act for ALS. And you'll see all the hard work that we've put in to advance the scientific knowledge of neurodegenerative diseases, push for innovation, and support the development of urgently needed new therapies for neurodegenerative diseases. Finally, I want to give a huge thank you to CPATH for convening today's meeting. I really appreciate all the efforts that you put in to bringing everyone together to provide these updates. I'll now turn the floor back to our meeting organizers. Thank you.
Thank you very much, Teresa. It is my pleasure to now moderate the first panel of the morning, where we will hear perspectives from two members of the ALS lived experience community, Lynn Braumeyer and Kathy Collette. If you want to go ahead and turn your videos on, we can get you spotlighted here too. Okay, thank you. Tanya, I think we can probably stop sharing screens while we launch into this conversation. So first, welcome, Kathy and Lynn. We are so grateful for you being here and for the opportunity to really anchor today's conversations in the perspectives of two leaders in the ALS community before we transition into our research updates. So kicking us right off, I'll ask each of you to briefly introduce yourselves and share a little bit about your lived experience with ALS. Lynn, maybe I'll ask you to go first.
Hello, welcome to this discussion. I am an engineer with ALS. I'm now in my seventh year, seventh year. To back up a little bit, there are roughly 30,000 of us at any given time. For those not familiar, I'm sorry, hold up a minute. Go to Kathy.
Sure, absolutely. We'll put a pin in that, Kathy.
Thank you. Thanks very much for inviting me to be here today, Megan. We lost Mom to ALS almost 30 years ago. And at the time, I found it outrageous that she had the same prognosis that Lou Gehrig had in 1939. And I think it's all the more outrageous today. My passions are better clinical research and better public policy. I have some background in the pharmaceutical industry, some science background, and a lot of business background. And quite honestly, I have a lot of unfinished business with this disease.
Thank you, Kathy.
I fixed
Yeah.
my
Okay.
audio. I was saying, for those not familiar with ALS, there are roughly 30,000 of us. We're a rare disease, but not an ultra rare disease. Roughly 50% of us die within 3 years of diagnosis. Lou Gehrig died 2 years after diagnosis. Roughly 90% of us die within 5 years. I'm at 7, so I'm essentially an ultra rare subgroup of ALS. In clinical trials, we are now routinely excluding people 24 months from symptom onset. Lou Gehrig was a graduate of Columbia, and they had a large medical hospital even in the 1930s. He could not be diagnosed there. He went all over New York State and Boston unsuccessfully, had to travel to the Mayo Clinic in Minnesota to be diagnosed.
Yeah.
Even today, diagnosis takes a long time, often a year or more. So the concept that we're cut off from clinical trials
Yeah.
24 months from symptom onset would not be accepted in most rare disease communities.
Yeah.
I'm seeing a lot of frustration in all of my social media feeds about a lack of clinical trial options. That's why reauthorizing Act for ALS is so important. In the past 5 years, the majority of that money went to extended access protocols, EAPs, and that's all that those of us that don't qualify for clinical trials have. Thank
Thank
you.
you for highlighting some of those things for us, Lynn. That's actually a perfect segue to my next question, which was going to be around really I was going to ask you to share a little bit about the real grassroots effort that led to Act for ALS. So recognizing some of the challenges that you have just identified. Kathy, I'm going to turn to you. I know you were you're part of that movement, that grassroots movement that really led to authorization of Act for ALS initially. I'm wondering if you could tell us a little bit about what motivated that work and where it came from. What were the things that the legislation was trying to address, particularly with regard to coordination across the research community?
There, about 6 years ago, it was right before COVID. We had some what I would call rock stars in the ALS space, people with ALS and caregivers who were what I call good troublemakers. And they had started asking some questions at the time. And some of the questions they asked related to things that were going on for other diseases, especially things provided by NIH. We looked around and saw huge natural history studies in other disease areas, but in ALS, we had small natural history studies that really weren't big enough for a heterogeneous disease. So natural history studies seemed like a huge need. We saw things going on with NINDS and Parkinson's and the AMP-PD getting the private sector involved. And it's like, that seems like just a compelling idea for a disease like ALS. And then finally, this is a little off that topic, but we were all going to seminars and things about EAPs, but you look around and there weren't any for ALS. And it's like, what's the problem here? So as a result of all of those questions and trying to come up with some answers, Act for ALS emerged. And it emerged to give NIH the tools and resources that it needed to really step up for ALS. And I am grateful that they did.
Thank you, Kathy. And you both have been involved. We heard from Teresa earlier that the lived experience community has been a key part of the work that's ongoing as a result of that act. Can you share a little bit about why it's important to you to be involved, how you've been involved, and why you feel it's important? Lynn, we'll go to you next, and then to Kathy.
As I mentioned, I have not qualified for a clinical trial in over 5 years. EAPs represent a glimmer of hope for roughly 22,000 of the 30,000 of us before Act for ALS. Only about 100 people got EAPs. Currently, about 1,000 have got EAPs. That's well short of the total population of us grasping for straws. It's important to remember that research in general is crucial, and some notable progress has been made reverse engineering a couple of rare genetic forms of ALS. But we still don't have consensus on the biomarkers needed to be able to categorize all of the subpopulations in ALS. And EAPs in particular help to do that, because we're looking at people and possible impacts to outright survival, which is crucial in a fatal disease. Thank you.
Thank you, Lynn. Kathy, can you offer your perspective on why you think it's important for the lived experience community to be engaged
Well,
in?
and people with lived experience, when you've been a person with ALS or caregiver, you have given up a lot to ALS. And as far as I'm concerned, you have standing in any group that is discussing the fight against ALS. So I think that's important. And I think that one nice thing about what has happened with the public-private partnership is that I think some of these groups gives us a peek into areas of research that we hadn't had a peek into before. And my hope is that they'll listen to us in case we have some ideas that would be helpful.
In our 1 minute remaining, could each of you offer your top priority for what the research community should focus on moving forward, what we can do better in your view? Kathy, maybe you go first.
I'll just jump on. We've accomplished some things, but you can't call it mission accomplished by any means. I mean, we have accomplished some really good things. I think that there are also some opportunities, and people at NIH have heard this from me before. So I think we need to move faster. You can't say we're going to let a deadline slip a quarter, which you would sometimes do in business without thinking twice, just because you're dealing in quarter status reports. But you have to move faster. You have to be very conscious of the ALS clock. I think we still have silos. And Colin and CPAP have done a heroic job at taking data from our silos and making the data aggregated in a form that's useful. But sometimes I think we just need to get rid of some silos, maybe consolidate some work. So, and as much as we talk about being patient-centric, I think we can be more patient-centric about the way we gather data, especially for multiple studies. And I think we can be more efficient and put ourselves in a better position for the machine learning age. So just some thoughts about where I hope we go.
Thank you, Kathy. All right,
In a
the last 20
disease,
seconds go to you.
in a disease where most of us die within 3 years, we need to do a better job of getting preliminary results out. Right now, people are holding data close to their chest for a variety of reasons, but especially thinking about the reauthorization of Act for ALS. This is handicapping our ability to influence research funding. Thank you.
Thank you so much to you both. We really appreciate you joining us today to offer your perspectives, and thank you for all you are doing for the community. It's a joy to work with you. And with that, I will pass it back to Tanya to introduce the next session. Thank you.
Thank you, Megan, and thank you, Kathy and Lynn. Our next session is entitled Updates from FDA and CPAD, Building a Trial-Ready System for ALS and Other Rare Neurodegenerative Diseases. We will first hear presentations from our FDA colleagues, Dr. Michelle Campbell, Associate Director, Stakeholder Engagement and Clinical Outcomes, and then from Dr. Catherine Needleman, Director of Orphan Products Grants Program. After the presentation, we will hear updates from our CPAD colleagues, Dr. Colin Hovina, Vice President, Rare Orphan and Pediatric Diseases, followed by updates regarding Huntington's disease by Dr. Tarina Martinez. At the end of those presentations, we will hear from Lauren Holder, who will give her perspectives as someone who lived with lived experience in HD. After the presentations, we will have an opportunity for audience questions and answers. Again, to our audience, please feel free, if you have any questions, to put those in the Q&A chat. But first, to our colleague, Dr. Michelle Campbell. Michelle?
Thank you, Tanya. Good morning, everyone, and thank you for joining us on a Friday in June to receive an update on what has happened over the last few years in the implementation of the Act for ALS from the FDA's perspective. Next slide. So as a quick refresher, if you haven't been implementing Act for ALS over the last couple of years, it was passed into law in 2021 with various sections. I'm going to be talking about the sections that I've highlighted that were tied to FDA's portion of what needed to be under our purview, which is parts of Section 3, which is the public-private partnerships jointly with my colleagues at NIH. Section 4 on the action plans we were to develop. And my colleague from the Orphan Products has been mentioned, Kathy Needleman, we'll be talking about the Rare Neurodegenerative Disease Grants Program. Next slide. So we were tasked and in a meeting and webinar that we presented in August of 2022 to put together an action plan for rare neurodegenerative diseases, including ALS, of areas to address unmet needs in this disease space. And also we developed an ALS science strategy to look at potential ways to address issues in current drug development landscape at that time when we wrote this almost 5 years ago. And really we developed a multi-pronged approach in implementing this action plan, leveraging existing programs that were occurring at the FDA at the time. And I will say going forward, as we've been implementing this, programs that have been developed since then, as well as collaborations with our colleagues at NINDS, and really leveraging the public-private partnerships that have been created to support really the identification and the advancement of our needs that we have in rare neurodegenerative disease drug development, including ALS, and what have we learned from that, and what do we hope for going forward. Next slide. So some of these slides, if you may remember from 2022, I pulled these back out to refresh us about what we were hoping to do. Still available online on our website is the entire action plan. But we knew we needed to think about where was the direction of where science was going in rare neurodegenerative diseases and what could be achievable with the resources that we had. And so one of the first things that we have done is that we have established at FDA an internal task force or group across all of our medical product centers and the Office of Orphan Products to advance the drug development needs in rare neurodegenerative diseases. This group meets quarterly to discuss ongoing things. When it comes to rare neurodegenerative diseases, we present maybe recent approvals that have happened in rare neurodegenerative diseases, innovative methodologies internally, and asking ourselves from our internal experts, how can we leverage information from our other medical product centers that could be informative to our own advice that we may be giving to industry who's coming in and asking us questions through the IND programs. This has really been a great opportunity to formalize something that we already were doing in an unformalized way, which is talking to our other center partners on a more formal and regular basis. But I will say that we meet all the time on an ad hoc basis when real burning questions come up, and we don't wait until that scheduled meeting. We are constantly talking to them on a regular basis and trying to solve problems jointly together that may be impacting our centers with the development programs that we see. The next aspect of the action plan was the development of the public-private partnerships, and you'll be hearing more about them in the next few presentations. And really that is, I would say, a true hallmark of the efforts that have been pushing forward and identifying and working together collaboratively in a pre-competitive space to looking at leveraging data from multiple data sources, conducting studies and observational studies and data analyses in ways that are really pulling all those silos, as Kathy and Lynn mentioned, together and trying to pull and understand better the science, where it is, and identifying gaps. And you will be hearing about examples of where we have identified really critical gaps in the communities of where we're missing important information and how can we actually fill those holes. And one of those efforts is happening under CPR&D right now in the ALS community with trying to do the needed qualitative work of really truly doing understanding of their lived experience and how can that be informative into endpoint selection in our clinical trials. We continue at the FDA to support external engagement with our rare neurodegenerative disease communities, and I would say with all of our communities. Our patient listening sessions and our externally led patient-focused drug development meetings are a continued success from these programs. I would be remiss to say that I feel that almost all of the sessions that we participate in out of the Office of Neuroscience, as well as my other two medical product center colleagues, typically are in the rare disease space, and most often they are rare neurodegenerative diseases. This engagement that we partake in is critical to us for our everyday work as reviewers. It's informative for us to really hear directly from patients as they are experts in the diseases and disorders of what that lived experience is and what their hopes are and what would a benefit look like to them. We find this information extremely informative to us, and we do take that information back and incorporate it into our everyday review work. And I know that can be hard to see sometimes because that review work, the ultimate product of success, would be a potential approval of a marketing application. But do know that we do leverage this work and think about it when we are providing advice to development programs. And I would say we've definitely leveraged the success of that, and it's only continuing to grow with our rare disease communities and the engagement opportunities that we have. Some other goals we had is we did develop an ALS science strategy at the time of the initial action plan. Our goal had been to develop more, and unfortunately, due to constraints, we were not able to. But I would like to say that did not mean that we did not identify other disease areas where we could apply science strategy ideas without having a formal strategy and plan. And one of those would be in Huntington's disease. And you'll be hearing about some of those activities that are currently occurring to address the current unmet needs in that area. I think we do hope that if we are able to see the next round of this, we will be able to do additional science strategies for other rare neurodegenerative diseases, because we do know that they are important to communities. But I do want to emphasize is that we still do consider and think about those unmet needs and try to leverage through our existing pathways. How can we build those unmet needs science strategies and other potential opportunities to do research and to gather information and provide advice? Next slide. So in the ALS science strategy at the time, I would say we have accomplished a lot of these goals. First and foremost, as I mentioned, was this working group for neurodegenerative diseases and with a sub-focus in ALS, and that has continued in happening. And we've been supporting translational science through efforts through the public-private partnerships. In addition to efforts through the Rare Neurodegenerative Disease Program, as well as efforts with NINDS, we are exploring the gaps of the ALS natural history and how that has evolved over time. I think we have definitely learned that people living with ALS are living longer, and we have a better understanding of really the true natural history of these people, so we can be able to give better advice and think about what drug development should look like. We have continued to encourage and facilitate data sharing, not only through CPATH, but through FNAH with the data knowledge portal that you'll be hearing about. Efforts through CPR&D has been looking at developing data standards, as well as standard operating procedures and analysis plans. We have a continued focus in the exploration of digital health technologies. I think we have said many times that we think that digital health technologies can help us address some of our big unmet needs in the neuromuscular communities and with ALS. Work has been happening on multiple prongs with this through CPR&D and looking at digital health technologies in ALS. We continue to encourage sponsors to include expanded access into their development programs. And as you know, FDA is extremely supportive of expanded access programs. And 99% of the time, we typically are able to make those decisions. So we will always continue to encourage expanded access. And obviously, I think the efforts that you'll hear in the public-private partnerships for more bench-to-bedside efforts is underway. We will always continue to explore innovative trial designs, and we've been putting on various webinars, not in the rare disease communities and meetings, to talk about innovative trial designs and the applications. And I think that is still going to continue on and be forthful. We understand that trial infrastructure and the abilities to conduct clinical trials in our rare disease space, but particularly in ALS and our rare neurodegenerative diseases, is always needs to be evaluated. I'm going to be sharing next an effort that kind of looks at cloud infrastructure and patient burden and looking at opportunities to maybe decentralize aspects of clinical trials. So I do think that we have been as successful as possible with the resources we've been given to implement as much as our action plan and our science strategy over the duration since the initiation of the Act. And I think we look forward to continuing to update and advance what we've learned and be able to apply those to other diseases. Next slide. I want to quickly highlight, you can go ahead to the next slide, Tanya, an effort where we were looking at remote administration of the ALS FRS. So the ALS FRS-R is typically used as part of the primary endpoint in our ALS trials. It is typically a clinician-reported outcome that the assessment would need to happen in clinic. Obviously, during COVID, we saw an influx, and not just in ALS, but most diseases, of trying to move things to be done at home. And while we knew that, we had heard things that the ALS FRS could happen at home and may have been, we really needed to make sure that there was a standardized way of making sure the data was being collected and administered in a standardized approach, but also that the results of an at-home assessment would still be interpreted the same way if it happened in clinic. Next slide. So through the Office of Orphan Products, we were able to fund a contract and do a study that actually looked at remote administration. One of the products of this has been a literature review that kind of scoped what was currently known in the literature at the time and was published in 2024. Next slide. Additionally, in this research, we engage patients and advocates to guide the implementation and development of this protocol and understand the technology capabilities of families. They were instrumental in helping develop the training materials. They offered a pilot, and then we did a comparability study. I will note that our patient advocates represented 12 different time zones. It was a global effort in terms of having that input, and they truly were critical to the success. In the comparability study, we looked at the administration of in-clinic, a telephone-based, and a web-based video platform to assess looking at the administration of the ALS FRS, as well as patient and caregiver satisfaction of having which mode did they really prefer. So next slide, and I'm going to skip a little bit quicker because of time. One thing that was developed, leveraging existing known information, was a standardized training for remote administration at the ALS FRS, and this has been made publicly available. Next slide. Let's go ahead to the next slide, please, Tanya, for time's sake. So in our comparability study, we did a nationwide study of 7 sites, trying to get as diversified population as possible, with 3 dyads that had a crossover between either in-person or one of the modes of remote administration. Next slide, please. So as you can see, this was this research scheme of how the administration happened. And this was a short crossover-based study looking at comparability, as I said. Next slide. And so really what is the take-home message in this slide is that when looking at the scores of the ALS FRS among the different modes with the in-clinic piece, we still saw good correlation among the modes, and that really said that we could be actually doing this and still know that the results would be maintained and interpretable. Next slide. What we also found was that the ordering in which the occurrence of the mode of administration, so if you first started in clinic and then did it at home and then continued back in clinic, or you did it at home and then went to clinic, that we could still trust those results. There was no interpretation issues. It's the one day you're really happy that a non-significant result does occur. It was very exciting to see that. So this is known as sequence effects, and we are really happy to see that nothing happened. And so there was strong evidence to support all 3 of those modes of data collection. The next slide. We did find that patients definitely thought, and caregivers were very happy with the administration and liked the remote option, but they did also note that they also like having the connection to their care site, to the clinical site, and still wanted to make sure that was an option. So I really felt that this was a really good way of saying there are families who may be okay with doing a remote visit, and there may be some that may still want to be a clinic, and that option should still be made available to them. Next slide. So a manuscript is to be submitted for publication of these results. All of the data from the study has been data shared and contributed to RDCA DAP, and that we can start providing advice to sponsors if they ask about decentralized trials in ALS, what are the considerations and what our thoughts are from this study. Let's continue on to the next slide, and I'll be wrapping up shortly. I also want to highlight efforts from our Center for Devices on implantable brain-computer interface devices, which has been an up-and-coming area with great potential use for ALS. And so efforts in this area has really been looking at the alignment of COAs, clinical outcome assessments to help demonstrate clinically meaningful benefit and target patient populations, and this is including ALS, and also alignment on what a clinical trial will look like, including the COAs used, the sample size, inclusion and exclusion criteria, and actually the duration of the trial. So CDRH is extremely supportive in working on these efforts. Next slides. And one of the ways they've been doing that is been through, if we can go to the next slide, please, has been through working on consensus building to support future COA development with BCI devices, and they've published a landscape analysis work that was conducted through another grant from the Office of Orphan Products. And they are engaging with stakeholders through a collaborative community to support this development. So they are actually also an area that we often may not think about is what our certain devices can help us and do when it comes to treatment options. for rare neurodegenerative diseases. And this is a great example of how they are also doing work to help in this space. So my final thoughts are, and if we go to the next slide, so I can turn it over to Kathy, is that we continue to support the advancement and development of medical products for rare neurodegenerative diseases. We are leveraging our existing rare disease efforts to support our action plan, and we hope to be able to expand to more diseases in the future. So I'm now going to turn over to my colleague, Kathy Needleman from the Office of Orphan Products, who's going to talk to you about the great work that they're doing.
Thank you, Michelle. I appreciate all of that. Today, I'm going to talk to you a little bit about our programs. I'm going to jump back a little bit into the brief background about our office and about our programs, talk about the clinical trial grant program, as well as the natural history grant program, and of course, get to the rare neurodegenerative disease grant program in terms of what has been done and what fiscal year 26 looks like. Next slide. So our office supports and provides incentives for sponsors to develop products for rare diseases. The real mission is to promote the development of drugs, biologics, and foods for patients with rare diseases. We have several designation programs as well as several grant programs. All of these, the designation for programs, depending on where they sit, are providing incentives to move research along in rare disease development. Similarly to the grant programs, to provide funding and support for these purposes as well. And a lot of this is on our website, so feel free to look at that at another time. Next slide. Our program was established back in 1983. The Orphan Products Grant Program is really the umbrella program that provides the funding for clinical trials grants as well as natural history grants. It's really here, it has a budget of about $19 million this year, and it advances the development of these products that demonstrate products for the diagnosis and treatment of rare diseases. The clinical trial grants, we have we have about 75 ongoing studies at any one time, sometimes more, sometimes less. The focus is really on efficiency and innovative trial designs. Our grants have led to over 85 product approvals, as well as numerous publications and multiple impacts and regulatory decisions in the field. In terms of the natural history grants, this was launched in 2016 after seeing a real need to really understanding more about these disease progressions, really being able to inform better clinical trial development. And currently we're funding 7 grants, although we are within a new receipt date. We're just wrapping up a cycle. This last cycle for fiscal year 26, we had one of the largest application receipt dates. We had 90 applications come in, but notably, even though this wasn't specifically for rare neurodegenerative diseases, 11 of these were for actual rare neurodegenerative diseases. And these studies have potential impact for clinical trial development, supporting regulatory decisions, as well as collaborations. We see numerous collaborations through this program with industry and patient groups, which lead on to moving products along and publications in this field. Next slide. This gives you a little historical view of our program in terms of funding. Back in 1983, when the program was started, we had very little funding, very little authorization as well as appropriations, and we've grown over the years. Right now, our authorization for our program is of around $30 million. And as you can see, we're generally appropriated for less than that. However, we do get some additional funding through the centers as well as other support that allows us to do more work in these areas and fund additional grants. Next slide. Since 1983, we have funded, we have seen over 3,100 applications and reviewed those. It's led to over 800 studies being approved, and we are funding quite a number of research in this field. There are over, the real take home here is that all of this sort of boils down to being able to lead to product approvals, which is our main mission. And we've seen over 85 products approved that were at least partially funded through the program that's helping over 90 indications in the rare disease space. So generally around 10% of our funded studies do lead on to marketing approval. Next slide. This slide gives you a little bit of a representation of what we fund. We have funded multiple disease areas, and generally our largest disease area tends to be oncology, although that is a combo, so it's general oncology, heme, neuro-onc, it's combined. You can see we have funded in the neurology space about 107 awards, where 36 of those actually were for rare neurodegenerative diseases. And then you can see the plethora of other diseases that we do fund. Next slide. Prior to the Act for ALS, I want to talk before we actually started seeing additional support for research in this field. We were funding rare neurodegenerative diseases in that time frame. So there were about 32 clinical trials over that time that totaled over $27 million for rare neurodegenerative diseases, a small percentage of the overall time there, since this program was moving on since 1983. In that prior time, we had 3 natural history studies, and actually there were 2 approvals that were partially supported by our program that are listed. Next slide. But as you heard earlier, since the enactment of the Act for ALS that was established back in December 2021, and with the real purpose of having some, through Section 5, grants and contracts to support and cover research on the development of interventions to prevent, diagnose, and mitigate, treat, and cure ALS, as well as other rare neurodegenerative diseases for adults and kids, we were able to do much more. Next slide. So since that time frame, so in 2022, which is the first fiscal year, we started funding, specifically directed funding for this area. That particular year, we had a natural history cycle, and 3 of those natural history grants were supported through this program, 1 for ALS and 2 other rare neurodegenerative diseases. In fiscal year 23, we were able to take on another 5 studies for natural history. And then in fiscal year 24, we were able to take on an additional 1. So as you can see, just since the development of this program, 9 out of the 17 natural history studies that we would have funded through our natural history grants program were actually for rare neurodegenerative diseases. Next slide. This slide gives you a little bit of an overview of what we actually funded in these different fiscal years. I'm not going to go through these in general. It's just nice to see who are being awarded, what they are looking at. As you can see in fiscal year 22, we had an ALS study and 2 non-ALS studies that we were being supported. And as Michelle just mentioned, those 2 contracts that were summarized earlier were funded in that fiscal year. What's notable in that fiscal year, there wasn't a specific amount of monies that was directly moved into this program. However, we were able to use an increase into their program to fund our neurodegenerative disease grants. But even better, we were able to leverage that money with our collaborations with NIH and elsewhere in the agency to really put in about $5.8 million for these projects to really start going in this new realm for rare neurodegenerative diseases. Next slide. This gives you the fiscal year 23 awards. As I mentioned, those 5 natural history awards are listed at the top. 3 of actually were for different aspects of ALS to really sort of look at different things that could help move that field forward. 2 for non-ALS indications. But we also expanded an ALS natural history study that we funded in the past, really to expand that trial and be able to get additional information that we felt was really needed for the regulatory space within ALS. So that, along with a clinical trial that we supported for familial dysautonomia that particular year, really rounded out our portfolio for rare neurodegenerative diseases. Next slide. And then in fiscal year 24, we were able to support an additional biomarker study for rare neurodegenerative disease and utilizing that with an addition of supporting what was already ongoing. So next slide. So after fiscal year 24, we sort of switched gears. When we went into this and thinking about a plan for our program, we wanted to ensure we're meeting the needs of the patients. We had talked to stakeholders, we've talked around the agency, and we really wanted to align with the FDA action plan. So at this point, we sort of switched gears into funding clinical trials and having directed RFA support for rare neurodegenerative disease clinical trial research. An RFA was posted and had receipt dates in the following 2 fiscal years. Next slide. The real purpose of this RFA was to really look for and fund clinical trials that would be evaluating efficacy as well as safety to help establish and support new indications or change in labeling for these rare neurodegenerative diseases. Eligibility, we fund pretty much anybody who's outside the federal agencies. Of course, you need to be a rare neurodegenerative disease, and defining that as such in your application. The budget for those last two receipt dates were about $650,000 in total costs for up to 4 years, but we offered an additional $250,000 to support innovative trials. We really heard the need to really move these products along faster, get results quicker, hear about what can we do to sort of emphasize that with our researchers. And so we developed an additional kind of a carrot or incentive to sort of come in for these adaptive designs or seamless designs, umbrella platforms, things like that that could really move the body of work forward quicker so we can get answers and then potentially move that along in phases of development. Also, we offered of additional funding for things like data assimilation or modeling that could also, with small, potentially less amount of work with patients, but more amount of work with understanding the safety and efficacy of a product. Generally, in terms of the number of awards we would fund, really depended on what we would be appropriated for those fiscal years. We expected to fund about up to 2 awards each of those years with the money that we had ongoing, but really depended on the quality of those applications as well as the availability of those funds. Next slide. So fiscal year 25, we awarded 1 application for AT. It just started last year, and this year, coming soon, we hope to announce those by the end of the fiscal year. We are just wrapping up that cycle now. Next slide. So kind of where this all and why and how this all built up from bottom up. So where we started in funding these, we were really looking for thinking about natural history and biomarker research, thinking about the collection of biosamples, biorepositories, things that will ultimately help further along in research development. Also looking at how those biomarkers could advance clinical outcomes, or how can we characterize those diseases. So really that's setting that strong foundation, moving up to really characterizing diseases, thinking about their phenotypes, thinking about COAs, building even further as we hear clinical trials and as our contracts have done, really thinking about how to decentralize that model. So even in different rare neurodegenerative diseases that could be utilized across different diseases, funding things that could really have impact to recruitment, e-consent, thinking about early diagnosis, thinking about baseline data acquisition and things like that. And then really developing those novel biomarkers, setting that stage for early detection and making sure we can find these and detect these early. So all of the studies that we funded sort of fall into sort of that realm. And then as we came up, we're now funding our clinical trials with those innovative and efficient approaches, as I mentioned, even seeing things like individual ASO therapies and really seeing how this all can fit into the construct of moving rare neurodegenerative disease research forward. Next slide. So thinking of beyond this year, we use those concepts that were obtained from our stakeholders to establish our funding opportunities to meet unmet medical needs in this field. We are moving along, and things then for this fiscal year will be funded by September 30th, 2026. We are looking ahead for the reauthorization of the Act for ALS. If that comes into fruition, we're certainly already thinking about what could be done, future directions to have the most impact in this field and really build on what already we know and the knowledge we've gained through the current studies. Next slide. And so quickly, conclusions. We've been successful in helping with product approvals, publications, getting data out there, helping regulatory decisions and thinking about standard of care changes. Truly increasing collaborations. What we've seen a lot with our natural history studies is that real direct collaborations with researchers and industry really partnering with these people after they've gotten funding through us and really seeing how they can use that data in their clinical trials moving forward. So that's been a real opportunity in this field. And then we still see a high need for quality clinical trials as well as natural history studies in this field. And we also want to make sure that whatever changes we make will impact the patients and the most needed work that needs to be done. But as we know, there is a lot of work to be done, but working together is the best way to get there. So I'm happy to be part of this meeting today, and thank you for the opportunity. I think that's it. Next slide. We do, just back one, sorry. There are RFAs out there still. Even though these are our clinical trial and natural history studies, we do have an October receipt date for those researchers interested in clinical trial research. And we have we just did a natural history receipt date, but we'll have another one in February 2028. So things to keep on your calendars if you have researchers interested in funding. Next slide. And that is it. Thank you.
Hello. I want to thank everyone for attending and echo a lot of the sentiments of people that have previously spoke, and just the welcoming of the community to people new in ALS. I always tell Kathy I'm ALS adopted, and I think that she's been one of those people that have really been an awesome person to work with, and Lane has taught me quite a bit. I wanted to open the discussion today around just a bit of the history. I'm going to quickly go over this and what CPATH is, and then move into some of the work that we've done. But just so everyone knows, CPATH has been around over 20 years. Our involvement with as one of the PPPs for this initiative, SCPR&D, is in September, and that's actually when I joined CPATH. I was joined and recruited, and it was sort of the first thing that I started doing. And so the community and the engagement that I learned in this was just amazing. And I'm a bit sentimental today, but also, energized to move into the next phases of this initiative. CPATH, we don't are not thought of, and we don't do things traditionally what you probably are familiar with a nonprofit. We focus on creating regulatory grade tools to help advance drug development. So obviously researchers do research, regulators approve drugs, NIH will fund and support research, and individually communities end up taking those drugs. Our goal is to make this whole process easier, and so we look at refining those. And so we're one of the public-private partnerships, and our role really is to help translate the science into things that will be actionable, meaningful, and in improving treatment-oriented studies. Next slide. This is a slide that I want to emphasize. One of the things that was common when we first started this is that how do we all play well together? How do we make and connect? And fortunately, the community that was established under the Act for ALS was really groundbreaking, in my opinion. We share across working groups. We learn from each other. We try to understand how to move through the science and the discoveries to better benefit the communities. I want to highlight, and you're going to hear this throughout the rest of the conversations, is that persons living with ALS are really instrumental to this initiative. They've been our North Star, and they participate, even given the short times that they might have, to guiding our process and informing things that how we develop the solutions. The projects that we're talking about today really aren't things that CPATH generates ourselves as the idea. There are things that are defined under an unmet need development process where we go out and talk to, persons with lived experience, pharmaceutical companies, regulators, researchers, clinicians, all based on that feedback. We develop what is our scientific strategy. So what you're going to hear today is some of those elements and how they impacted over the last years. Next slide. Now, I'm just going to talk about a few priority areas. This isn't comprehensive. I'm going to talk about the top group there. The questions that we're asked is, how do we streamline access to standardized high-quality data for stakeholders? And particularly for including clinical trials. How do we make better decisions? This data, obviously, making it readily accessible, but also usable in ways that it hadn't been done before. And you're going to hear about that through the conversations today. Improve clinical outcome assessments, resources to standardize clinical outcome assessments. One of the things that was really paramount in this was there was a lot of new companies and a lot of new researchers in this space, but without the experience. And one of the things that they needed was things that would help them onboard and whatnot to be able to be more successful and be more efficient, because we realized time is very important here. evidence to support digital health technologies, or DHTs, as biomarkers and clinical outcome assessments for ALS clinical trials. And then, understanding the evidence of how novel biomarkers might inform regulatory decision making. We've seen approvals happen during the time of the Act for ALS. How can we begin to learn and expand for those? And I'm going to highlight actually in the top ones, and then let Shubhangi talk to some of the other pieces. Next slide. What I'm going to highlight here, you already heard about RDC Adapt. That's our data platform. The knowledge portal was established with the idea to meet the aim of what was represented in part one. CPATH had a long history of using and curating clinically based data from natural history studies, registries, and clinical trials in ways that would be more readily adoptable from regulators. And so our role was to really focus on whether the clinical data pieces that would be ultimately linked to the ALS knowledge portal. Shubhangi will talk more about SAGE later on today, but this partnership allowed the collection and aggregation of data aligned together in that one-stop shop that people wanted and requested as part of the initiative. And as we sing this, you're going to see a manuscript that's coming out probably towards this fall, but a big meaningful element to how and what can be done with the portal, what data is there, and some examples, use cases, and whatnot. Next slide. Now, RDC Adapt, one of the things at RDC Adapt, this is a project that was in part funded by FDA as its core, but also additional funds to focus on rare neurodegenerative diseases. And obviously, we're one part of this knowledge portal, but we didn't stop just at this. And other initiatives that we do at CPATH is that good high quality data has been recognized as really important in understanding drug development. And so one of the things that's very strategic and appreciative in if you understand like regulatory alignment is the idea that you can learn from other diseases. And so one of the things that, as we moved into initiatives in ALS, we also began to look at similar diseases and also other rare neurodegenerative diseases. And so I'm happy to report we have 14 data sets in ALS right now, including the RTI study that Michelle mentioned. We're getting a Dr. Walk's study, which is also FDA supported. But we're also building data warehouses in FTD. We have one in PSP, and we have lots of data in ataxias. Trina may mention about this in Huntington's later on, but this is a wealth of data that we can begin to explore and learn things across rare neurodegenerative diseases. And all this is curated, standardized, and accessible by stakeholders. Next slide. Now, the two things that we're talking about COAs, the things that were important here was to establish consistency, helping people that are new to this space, so that they could be a lot more agile, but also then refinement. The first thing I'm going to talk about is the consistency piece. And the goal of this was really to look at the noise that was in ALS and learn from the experience, because the clinical trial infrastructure had been growing. There was a lot of new people entering, a lot of new sponsors, a lot of new researchers. And so what we tried to do, based on companies that have actually went through the process, take some of their learnings, turn them into questions, and create basically resources that would allow future people that have followed the same path to be more successful. And so really two working groups were formed to start. This isn't exhaustive. I'm going to highlight the first two. One is an ALS FRS statistical analysis working group, where we convene a lot of just amazing biostatisticians from companies, from Healy platform trial, from around the world that really focus on ALS in particular. And then in respiratory function. And that one was chosen as a first start because we started to recognize the need for this based on sponsors' feedback. There were sponsors beginning to look at more respiratory measures in clinical trials. Their importance in natural history studies become more paramount. And so we really wanted to get in front of that because of the value of that as an area of disease. Next slide. Just a quick update. We've worked with multiple stakeholders to align the methodologies for statistical analysis and synthesized a paper around a publication that took that considerations. We're beginning now that we have a significant amount of data, to look at the performance of those hypotheses and modeling, and then begin to codify and make recommendations, also linked to the feedback that we see from persons with lived experience. Next slide. For respiratory measures, what we ended up doing was we started identifying core sets of measures beyond FVC and SVC, and particularly work closely with regulators to ones that they consider adept in clinical trials, and then created a priority list that we subsequently supported for deeper dive. We had multiple presentations by different stakeholders that conduct trials. We also did a literature review and summarized the experts. We're in the process now of developing the clear sets of recommendations to inform stakeholders about how to use these measures and ultimately how they can be analyzed. And this will be produced and codified in a reference library. We'll continue on under, we'll continue on to expand in this with other concepts of disease. These are the ones that were prioritized in the first round. Next slide. Now, refining clinical outcome assessments. This is one that I can tell you over and over again in conversations. This is one that people echoed over and over again as being important. Really looking at how we measure success of a treatment and for ALS. And we worked through, obviously, survival and ALS-FRS being the cornerstone. The idea with this was to collect other information from lived experience and to expand things that could be more meaningful to capture. And so I'm going to talk a little bit deeper about what that was done. We basically worked closely with FDA. Next slide. To develop and follow their roadmap then guidance on the PFD guidance and selecting, developing, modifying fit-for-purpose clinical outcome assessments. And the first part of this is we looked at identifying concepts related to disease that were important to persons with ALS. And we found a good number and identified ones that might be amenable to treatment. We are now in the process of conducting additional qualitative work, working with the PWLE community to further refine this knowledge. And then we'll work further to look at either, as I said, modifying, selecting, or developing new COAs. Next slide. We also looked at trying to determine what instruments are being used. You notice the ALS-FRS is not on here, but because we understood its limitations. But we did look at very closely the performance of these instruments on various elements. I'm not going to go through each of these columns, but one can say, if you look at the question mark X's and lack of check boxes here, there's a lot of missingness in their fit for use elements to most of the instruments that are being considered in ALS clinical trials. One of the things that I wanted to highlight for this community is that if you look at the columns on the left, you see a lot of missingness on elements that are really important to involving persons with lived experience input. And so we'll be working through that, and particularly engaging the community to really clarify that, and we'll work through this as part of the process. Next slide. Now, digital health technology was another one that was outlined and mentioned quite a bit. There was a lot of natural history studies incorporating these, a lot of pharma companies starting these in their phase 2 programs, but not a lot of understanding of how they could be used in the context of regulatory grade trials. One of the things that was very new for us was the ingestion of DHT data. So we had to create a pipeline, an analytical pipeline, to be able to do that, which is something that was exciting and really transformative for the platform. And then we've actually worked through collecting priorities with researchers and persons with ALS to identify consensus on measures that should be looked at for considerations of biomarkers and COAs. And we've already started looking at lower limb measures, and we're in the process then of transitioning between additional upper limb areas, and then trying to make the sets of recommendation and gap analysis for things of how COAs and DHTs could be used as COAs and biomarkers in clinical trials. We're very fortunate the community has been working in parallel with us to try to create more alignment in how future data is collected. And there's been a really strong dialogue in educating different researchers on how the design of the study and impact of the study would ultimately impact the data and its probability use in the context of clinical trials. Next slide. I want to acknowledge quickly just the co-chairs that we have there, our other partners there. I mean, we've gone through a lot over the last years and working through to move the initiative forward. All of the ALS and R&D communities that are involved in this initiative have been just generous with their time and their energy and support. And we want to acknowledge that. Next slide. And then I want to highlight some other initiatives that we're going to have, and you're going to hear from some of the stakeholders later on today under our panel. But Trina is going to talk about Huntington's disease, but she also leads the ataxia program. We have multiple initiatives that also exist. We've heard about RDCA DAP. We're working in closely aligned R&Ds at PSP. You'll hear from Penny later on today. We've been working on building a data warehouse for FTD. Friedreich's ataxia, we've got a significant level of activity going on there, and we have a pre-consortium right now. We're very fortunate to work with HSP and the PLS community, and they've been just wonderful to collaborate with. And we have a meeting coming up in June that we're gonna work with NIH to identify additional gaps in moving the disease force, very similarly matched to what's being discussed for under the Act for ALS. Next slide. But I'm going to ask you, I want to acknowledge members of the core team here that have really worked through things as this has unfolded. And I want to highlight and make a call out, because we're always looking at including and engaging different persons with lived experience. And so I want to point you to Megan, but if you have additional questions after the webinar, feel free to email us and we'll get back to you on that as soon as possible. Thank you. I think that's it for the slides, and I'll be passing it to Trina.
Thanks so much, Colin. I really appreciate everyone who's gone before me to provide such a wonderful framework for the efforts that are ongoing across the community and at CPATH. So I'm just going to jump into providing an update, as Colin mentioned, on efforts within the Huntington's Disease Regulatory Science Consortium. Thematically, I'm just going to outlay the problem, which essentially is that we urgently need better tools for Huntington's disease clinical trials. Also to provide the solution to that problem that is a work in progress. I'm not saying that we have this completely solved, but what we are working on are scientifically rigorous, regulatory grade drug development tools that can de-risk and accelerate therapeutic development in Huntington's disease. The how is through the Huntington's Disease Regulatory Science Consortium, which I will abbreviate as HD-RISC going forward, and the why is why we're all here, to address the unmet need and deliver these novel therapies that are robust, feasible, and meaningful to people with Huntington's disease. So moving to the next slide, please. I will first give a very brief background on Huntington's disease. It is a rare disease similar to ALS, but unlike ALS that has a combination of genetic and environmental background, Huntington's disease is a fully genetic inherited disease. It has a insidious progression and multiple components of pathology and symptoms, including cognitive changes, motor changes, behavioral and neuropsychiatric changes. There are some management strategies. There is a drug that is approved for the Huntington's chorea, the movement change that happens in Huntington's disease, but that's a symptom support. There are currently no disease-modifying drugs for Huntington's disease that would slow or stop this horrible disease. It is, as I mentioned, rare, and because of the way that the genetic component causes the disease, there can be very variable age of onset and variable rates of progression. But we do have a lot of understanding in the recent years about what is driving the early parts in the pathology of Huntington's disease, and that's what we are focusing on for additional drug development. So that's what I'll explain in the next few slides. Next slide, please. This is just the introduction to the HD-RISC, the Huntington's Disease Regulatory Science Consortium, as being a hub for advancing these efforts in improving HD drug development. This was a consortium that was founded in 2018. It provides the neutral convening site to bring diverse stakeholders across the Huntington's disease community into a neutral space to do the hard work to develop these tools. In the last few years, we've adjusted our research plan to respond to the wealth of information that came out around the Huntington's disease pathology and progression, and also advances in drug development, opportunities for gene therapies, antisense oligonucleotides, really specific mechanism-based drug development, and to converge that into how we can rise to the moment with our research plan to focus more on drug development tools that can fill the relatively empty toolbox for Huntington's disease clinical trials in the earlier stages of the disease continuum. So one of the important ways that we do that is to leverage the Huntington's disease integrated staging system as a foundation. I'll explain more on this in the next slide, but using this to help us understand in a data-driven way what drug development tools can be advanced for Huntington's disease. Next slide, please. So this is a little bit more of a dive into the Huntington's disease integrated staging system, which I will refer to as HDISS. This is a conceptual framework that incorporates genetic, biomarker, clinical, and functional endpoints. I mentioned that Huntington's disease is an inherited disease. So stage 0 is essentially the genetic mutation that causes the disease. Moving then into stage 1, this is where biomarkers and early mechanisms of pathogenesis are occurring, but there's no evidence of clinical signs or symptoms. Individuals with Huntington's disease in this stage would not be feeling the signs or recognizing the signs or symptoms of Huntington's disease, which means this is an opportunity for us to understand what is happening in the pathology before the clinical symptoms occur, and it gives us an opportunity to intervene earlier if we can really understand these areas with granularity. Moving then to stage 2, this is where the clinical signs and symptoms emerge and continue to progress. And in stage 3 is when functional changes are overt and observable. So we use this staging system as a framework to help us understand the disease better. It is also a tool that clinical trialists can use to inform what to measure in the different stages of Huntington's disease, depending on the clinical trial inclusion criteria. And what's really important is that if we can show that something is slowing the progression from one stage to the next, that would be really impactful for Huntington's disease and could help reduce the long-term illness and extend the lives of individuals with Huntington's disease. So we use this as a framework. On the next slide, I'll walk through how we apply this framework for additional work within Huntington's disease. So next slide, please, Tanya. Colin earlier talked about a lot of the work in ALS for biomarkers and for clinical outcome assessments. I just want to point out that I'm using the term endpoints here. Endpoints are clinical endpoints are specific outcomes or events that are measured objectively and quantitatively in clinical trials to determine the safety and effectiveness of a treatment. Endpoints can be either biomarkers or clinical outcome assessments. And the way the endpoints are applied in the trials is to inform regulatory decision-making around safety and efficacy. And so we're working on how we can take what we know about clinical outcome assessments and biomarkers in Huntington's disease and get them to the level that they can be used to inform clinical trials. And so this is a big challenge because like ALS and like other rare neurodegenerative diseases, Huntington's disease has a huge challenge in getting ideal endpoints for the disease because it is a rare, small population. It's heterogeneous in the age of onset and rate of progression. It's relatively slow progressing. It's not as fast progressing as ALS. So the time horizon of a clinical trial is difficult to show something that is changing and sensitive. So all these challenges are, important and need to be considered, but this is why we are working so hard to develop improved endpoints. Next slide, please. So this is what the work that we're doing around patient-centric, regulatory acceptable endpoints for early Huntington's disease. We launched a new strategic framework in the beginning of this year, specifically to optimize Huntington's disease endpoints. We are following the FDA's patient-focused drug development guidance to achieve these efforts. We're leveraging the deep expertise and diverse perspectives of all of our consortium, casting a very wide net to get all the experts into the room to help us move this forward. We are doing rigorous evaluation of the data and evidence so that we can really understand the sensitivity, validity, changes, and meaningfulness. We're engaging regulators early and often for advice and alignment, and we are incorporating feedback from the HD lived experience community. In just a moment, you'll hear from Lauren Holder, who can give you some perspective from the HD lived experience community. The impact of these efforts is to improve the ability to detect onset and changes in meaningful symptoms. And this will provide consensus alignment amongst drug developers, regulators, and the broader community for identifying endpoints in earlier stage Huntington's disease. Specifically, right now, we're focusing on an HD clinical scale that is, I won't unpack the acronym here, but it's the CUHDRS. And by focusing on this, it will help us understand how that might be applied as an endpoint in Huntington's disease in earlier stages. And this can help reduce uncertainty, inform regulatory decision-making, and overall de-risk clinical trials in HD. Next slide, please. One very important component, I mentioned the why at the beginning of the thematic outline. Of course, the reason we do this is to provide improved therapies to the Huntington's disease community. Part of this effort is the recent launch of the HD Risk Lived Experience Working Group. This was launched just very recently, a month ago on May 15th, International HD Awareness Day. This provides a forum for the HD lived experience community and advocates. It has bidirectional information and perspective sharing so that we can bring the research and science updates to the lived experience community, let them from the bottom up, grassroots, share their questions, concerns, feedback, make sure that what we're measuring is meaningful. This is important because we really do need to understand what matters to people with HD and integrate and amplify their voice in these initiatives. Then we can also, of course, communicate this broadly to the HD community. So the impact here is bridging the gap between the scientific and regulatory communities with people impacted by Huntington's disease and implementing the principles of FDA's patient-focused drug development efforts and increasing transparency and trust. So that is actually my last slide. I just want to thank everybody who's part of our consortium because this is the village and army that is doing the work and I couldn't do it without them and also with my team at CPATH. I want to hand it off to Lauren Holder, who is part of our lived experience working group in HD Risk, and I want her to share in her own words and introduce herself because her words are so very important and impactful. Lauren, thanks for joining us today.
Thanks so much for having me, Tarina, and for that wonderful introduction and everything that you shared. So good morning, everyone. Thank you for inviting me to join you guys today. My name is Lauren Holder, and I have experienced Huntington's disease from multiple perspectives throughout my life. I watched my grandfather and my father live with the disease. I served as my father's caregiver and legal guardian before he passed away. I'm also gene positive for the disease myself, have participated in clinical research, and have spent many years working alongside researchers, regulators, industry partners, and fellow advocates. Those experiences have taught me something important, and that is lived experience is more than a story to be shared. It is expertise that helps us build better science. One of the most encouraging changes I've witnessed over the past decade is the evolution of how patients are included in research. The field has evolved from doing research to patients to doing research with patients. When we talk about lived experience, we're not talking about collecting stories simply to make research more patient-friendly. We're talking about a source of expertise that helps us ask better questions, identify what matters most, and build tools that better reflect the realities of living with these diseases. Initiatives like the Huntington's Disease Lived Experience Working Group, which I'm so excited about, reflect that progress. They recognize that patients are not simply participants in research. We are partners who can contribute insights that strengthen the research itself. One of the most important ways lived experience contributes is by helping us ask the right questions. Researchers spend years working to answer complex scientific questions, but patients and families can help ensure those questions reflect the realities of living with disease. Patients and families often notice changes long before they appear on a clinical scale. They understand the trade-offs they are willing to make, the symptoms that affect daily life most, and the outcomes that would represent meaningful benefit. They are often the ones asking, are we measuring what is easiest to measure or what actually matters? Without those perspectives, we risk measuring what is convenient instead of what is meaningful. The quality of the answers we get depends on the quality of the questions we ask. Lived experience also helps us build better tools. Whether we're discussing clinical outcome assessments, natural history studies, digital measures, biomarkers, or benefit risk frameworks, patients bring a perspective that cannot be captured through data alone. As patients, we may not always know how to build a clinical outcome assessment. We may not know how to validate a biomarker, but we do know whether a tool reflects our reality. We know whether a measure captures something that truly matters in our lives. That perspective is not a replacement for scientific expertise. It is a necessary complement to it. When patients are included early and meaningfully, the result is not simply more patient-centered research. The result is better research. And finally, lived experience helps keep research connected to urgency. For researchers, progress is often measured in milestones, data sets, publications, and development programs. For patients and families, progress is measured differently. It's measured in memories preserved, independence maintained, and time spent with loved ones. Every data point represents a person. Every study participant represents a family. Every day research moves forward gives families hope, and every unnecessary delay costs something that can never be recovered. Whether we're talking about Huntington's disease, ALS, or another rare neurodegenerative disease, the truth is the same. Time matters. That perspective is not intended to create pressure. It's intended to provide purpose. Because when we bring together scientific expertise, clinical expertise, regulatory expertise, and lived expertise, we create a stronger foundation for meaningful progress. As we continue building partnerships across Huntington's disease, ALS, and other rare neurodegenerative diseases, my hope is that we continue moving beyond simply listening to patients and toward truly partnering with them. Lived experience helps us ask better questions. It helps us build better tools, and ultimately, it helps us build better science. It also helps ensure we never lose sight of why this work matters in the first place. So let's continue building a system where science advances, collaboration thrives, and patients remain at the center of every decision, because lived experience is more than a story to be shared. It is expertise. Thank you.
Thank you to all of our speakers and panelists thus far in our webinar. I would now like to invite those that have presented and provided a panelist discussion to turn on their cameras for our question and answer series, and invite Katie O'Keefe, CPR&D Scientific Director, to moderate those questions. Katie?
Yeah, thank you, Tanya. As Tanya said, I'm the Scientific Director for CPR&D. My name is Katie O'Keefe. Thank you to all the speakers and presenters so far. This has been just a fabulous discussion and I know it's quick, and we will get to break here in just a moment. And I am excited to see all the comments coming in. So maybe to kick it off, maybe to you, Colin, because I know you discussed this just very briefly on the slides, but maybe you can speak a little bit broader to CPR&D's efforts that go into other rare neurodegenerative disease areas.
Happy to. The areas that we work in, they have, as I said earlier, the scope of what we do there is defined based on the feedback we get from that community. So we're not a prescriptive organization. We're an active listener and an enabler. For PSP, for example, we're working on a clinical trial simulation tool. For FTD, we're building a data warehouse and cross our fingers and looking at maybe alternative control methods. For HSP, we're working with them to go through defining their unmet needs. Let's see, for ALS, we also take learnings from what we've experienced in other more established diseases and inform them, not for ALS, just ALS, but these other diseases as well. And so I'm happy to go into any additional depth if people have particular interest to their individual condition. The ataxia programs, we have additional consortium. We have CPTA, and then we also have things going on with Friedreich's ataxia. And Friedreich's ataxia, we're also doing a trial simulation tool, but the data on the platform has been used in first in disease drug approvals as an alternative control. And so there's a lot of promising projects and work that we do, and it's one of the things that really inspires you to CPATH, and I enjoy the activities and engaging those communities. It's what drives me, as a person working here.
Thank you for that, Colin. I appreciate that feedback. The next question I have, maybe I'll ask Michelle to come on for this one. Could you speak more broadly to FDA's goals and future directions in ALS and other rare neurodegenerative diseases, and how the different elements from data to outcomes and understanding disease biology fit within this overall strategy?
Yeah, thanks, Katie. Thanks for that question. I really do think that as we're seeing in the advancement of science, particularly in our rare diseases, particularly in our neurodegenerative diseases that may potentially have a genetic component to it, we are seeing a great advancement in the understanding of what we call the biology or pathophysiology of disease. And we know that that is where a lot of therapies are trying to be developed to actually target that and try to intervene as early as possible. And really we're balancing both a pediatrics world and an adult world. While the focus has been on ALS, which is primarily an adult disease, rare neurodegenerative diseases cross the lifespan. And those approaches have a lot of similarities, and then they do have some slight differences in how we understand it. I would say good natural history is going to be paramount and fundamental to how we really think about the understanding of disease, how we can develop good biomarkers that are measuring disease. Can they be used to show a change in a therapy, as well as the assessments that may be needed to support an efficacy aspect, to really be able to demonstrate that clinical benefit. So I think as we go forward, what we do in our everyday when we're reviewing rare neurodegenerative disease programs is leveraging our knowledge from across programs and across diseases and applying some innovative and novel ideas when we're providing advice. I think for the broader community, I think going forward is from what can we learn from what we've done right now, and where does it make sense in what would be applicable in other diseases. So we take those lessons learned and say, let's apply this methodology. In the case of Huntington's disease and the efforts that are going to be underpinning for examination of the Unified Huntington's Rating Scale, that has actually come from prior work that was done not only with the ALS, as Colin described, but in a broader neurodegenerative disorder with Parkinson's disease efforts, and we're trying to leverage that into a rare disease. So I think we're going to take past learnings to inform future direction, but I would say that we definitely need good natural history, good rigorous data collection, and data sharing to really help be able to build those models that are needed to help be informative of where the gaps are, as well as what is needed in that translational science aspect. I think collaborative efforts between all stakeholders, both our governments, between governmental agencies such as us at NIH, which we do work collaboratively all the time, as well as other stakeholders in medical product development, so drug developers, device developers, DHT, technology developers, as well as academics. And of course, the most important people in any of these stakeholders is our patients and their families and the caregivers, because that is our number one stakeholder, is ultimately them. And so we need to make sure that they're at the table and that we're collecting the correct information from them so we can make the best informed decisions as we go forward to develop medical product development programs.
Thank you for that, Michelle, and thank you for everyone again for your participation this morning on the call. I know I'm getting pressured to let everybody go to a break, but I would say that we're going to continue responding to questions in the chat. We've had lots of great questions come in, so we are recording those, and we will try to bring in some of these additional questions into our next Q&A session near the end of the call. But right now, I think we're moving to a 10-minute break, and we will, Tanya, I'll turn it back to you so you can give them the time frame for when we'll be back.
Thank you, Katie, and thank you to all again for our speakers and panelists. We do have a 10-minute break, roughly 8 minutes now, but we will return and resume the webinar at exactly 10:50. Thank you. Again, thank you for your attendance. We will now begin our next session, which is entitled Updates from NIH and FNIH, Collaborating to Accelerate Research in ALS. And we would like to welcome our NIH and our FNIH colleagues, starting with Amelie Gubitz, who's the Program Director for the Division of Neuroscience, National Institute of Health, followed by Shubhangi Lal, who is the Senior Project Manager, Translational Science from the Foundation for the National Institute of Health. Amelie, to you.
Thank you. So good morning, everybody, and thank you for the invitation to give a brief update about the NIH implementation of ACT for ALS, as well as our overall rare neurodegenerative disease portfolio. So next slide, please. Okay, so we can skip again. On this slide, I wanted to show you just an organizational overview of NIH and to show you how the National Institute of Neurological Disorders and Stroke fits into the picture. So the mission of NIDS is to seek fundamental knowledge about the brain and the central nervous system, and also to apply that knowledge to reduce the burden of neurological disease to all people affected by those diseases. And NIDS is the lead institute for ALS research at NIH, but I've shaded some of our partner institutes also with light blue to indicate that there are many components at NIH that are interested in ALS research. And I would say that our partner institutes focus on specific aspects of the disease of ALS. So for instance, the National Institute of Environmental Health Sciences is very much interested in unlocking environmental risk factors of ALS. I also wanted to note that NIDS is the lead institute for rare neurodegenerative disease research at ALS. Next slide, please. Oh, something happened with the colors here. Anyway, this slide shows how the research stages that NIDS is supporting to advance basic discovery to clinical trials. And NIDS is invested in all stages of this research pipeline. So we have a strong investment in basic biology to understand the pathobiology behind neurological diseases. We are also very much interested in preclinical research that tests therapeutic concepts. So for instance, this can include efficacy studies in models of disease, but also toxicology studies or bioavailability studies that are critical to move a therapeutic concept into clinical testing. We also have a strong interest in clinical studies, so that includes natural history studies, and you've heard from the previous speakers how important those are, as well as biomarker studies, and that may include imaging studies or also the analysis of biofluids from patients such as blood and spinal fluid. And finally, we also have an investment in all stages of clinical trials, and that includes exploratory early stage clinical trials, as well as pivotal trials that provide the critical data that the FDA requires to make regulatory assessments. And another really important priority at NIDS is workforce development. So we have a very rich program of fellowship awards and career development awards to train the next generation of scientists, both in the neuroscience, in the basic aspects of our mission, but also in terms of neurological disease research. And this is really sort of where the mission of NIDS sort of ends. We are not involved in any type of regulatory assessments and regulatory decision-making, and we also have no involvement in terms of insurance pricing negotiations. Next slide, please. So when I was asked to present about our rare neurodegenerative disease portfolio, I sort of tried to sort of wrap my head around this, because essentially we are funding hundreds of grants across many, many rare neurodegenerative diseases. So one of my colleagues, Gabe Gonzalez, he sort of looked at our grant awards from fiscal year 2024 and mapped what rare neurodegenerative diseases are covered by that research portfolio. And then he sort of generated this word cloud, and this sort of shows all the rare neurodegenerative disease terms that were addressed by these research grants. And the bigger the font size is, the larger the investment is. And so, for instance, in ALS, frontotemporal degeneration, Huntington's disease, also Charcot-Marie tooth, those were the areas where the dollar amounts were a little bit higher, but nevertheless, we have a really broad portfolio across rare neurodegenerative diseases. And I should emphasize that this is really sort of directed by the research community. So we rely on the research community to send us in these really sort of exciting and meritorious research concepts across rare neurodegenerative diseases, and that enables us then to move forward with grant awards. Of course, it depends a little bit on the overall NIH appropriations, but I would say that sort of our investment across rare neurodegenerative diseases, it's very robust, but there will be some fluctuations between fiscal years. And I also wanted to mention that regarding my previous slides, in terms of the research pipeline, I think our largest proportional investment is in understanding the basic biology behind these diseases. And certainly this is a really important area for NIH to cover, because industry then is helping us to sort of do the later stage development regarding clinical trials toward regulatory assessment. Next slide, please. So for the purpose of today's presentation, I wanted to highlight two very exciting and very collaborative programs. One is the Rare Diseases Clinical Research Network at NIH. So multiple components of NIH participate in this program, and this is why it's not only just for rare neurodegenerative diseases, but for rare diseases generally. And the program is led by the National Center of Advancing Translational Research. So currently we are funding a network of 21 research consortia, and they have the shared goal of advancing clinical trial readiness. And what do we mean by that? So this is what I think Michelle and others have alluded to. It's sort of the refined understanding of the natural history of these diseases, the development of markers of disease progression, but also diagnostic markers, and then also the refinement of clinical outcome assessments. And another really important goal that is shared across these consortia is also training the next generation of rare disease clinician scientists. So I have marked the 5 centers that are addressing rare neurodegenerative diseases with red circles. So for instance, we have one consortium focusing on myasthenia gravis, one on hereditary spastic paraplegias, there's one on ALS, and there are also others on lysosomal storage diseases that also have neurodegeneration as symptoms. Next slide, please. Another program that I wanted to highlight is the Ultra-Rare Gene Therapy Network, or in short, the URGENT network that NNDS is leading. And this is led by my colleagues in our Division of Translational Research, who have a lot of know-how in drug development. So the goal here is to advance discoveries into clinical trials for people living with ultra-rare neurological diseases. And this program is quite unique because it does not only provide funding to academic teams who are developing these type of treatments, it is also providing access to expertise through contract research organizations as well as consultants. So for instance, the academic teams are connected with companies that can conduct toxicology studies, or also with consultants who have a lot of expertise in drug development. And the program really builds on recent advances in gene targeting treatments. So this includes next generation gene therapies and also agents called antisense oligonucleotides or small interfering RNAs and other agents. And they all have the ability to modify the expression of specific genes. And since many of the rare diseases are monogenetic, this holds really a lot of promise. So currently we are funding 7 active programs. One, for instance, is focused on ALS. That program is called Silence ALS. It is led by a team at Columbia University. Dr. Neil Snyder is the leading principal investigator, and they're really building on the success of tofersen, which is our first approved treatment for SOD1-linked ALS. It is using a similar technology, but it is customizing it to other ultra-rare forms of ALS, and that program is going extremely well. And then we also have other programs, for instance, in prion disease or in a lysosomal storage disorder, and then also across other rare neurological diseases. Next slide, please. So this slide just is a snapshot of the NIH research investment in ALS research since 2014. And as you can see, over the years, we have really a robust increase in our spending on ALS research. And I think that's really a reflection of the momentum in ALS research and also the excellence of the research community that is submitting really competitive applications to NIH. So since fiscal year 2022, we also have been extremely fortunate to receive additional funding through ACT for ALS. And as you all know, that funding is set aside for specific research purposes. Next slide, please. So this slide is just a recap of the NIH provisions within ACT for ALS. So one of the provision is Section 2. So this is our new research grant program on expanded access studies. This program is up and running. We are funding a number of expanded access studies right now. And this spring, we were able to release another request for applications. There are 2 due dates. The first one is in June, and we are hoping to receive another round of exciting applications. They will be reviewed this summer, and hopefully we can make additional grant awards for expanded access studies and before the fiscal year ends. And I was really happy that Lynn talked about this program from his perspective. So the importance of this program is that it provides access to treatment as well as research for individuals who are no longer eligible to participate in regular clinical trials in ALS. So for today, I was asked to focus a little bit more about Section 3. So this is our HHS public-private partnership. And you've already heard presentations about CPR&D by Colin and others. The next talk today will be from Shumangi Lal about the new Accelerating Medicines Partnership for ALS. And I'm going to focus a little bit on All ALS, which is our new natural history study in ALS. And as Colin and others mentioned earlier, we believe that people with lived experience are really well integrated and represented in this overall program, and all of the data is being shared with the research community through a new data portal. Next slide, please. So this is an overview of All ALS. It's a very large natural history study. It's really a great infrastructure, and I've just added the names and the headshots of the leading investigators at the 2 coordinating centers. So this is James Sperry at Massachusetts General Hospital, and then Robert Bowser at the Barrow Neurological Institute. So All ALS has 2 clinical protocols. One protocol is the ASSESS study. So this is a natural history study in people who are currently living with the disease. And there is a patient arm and a control arm. And the control arm here is really important for our biomarker studies. And then the other study is PREVENT. And this is a study that is enrolling individuals who are at high genetic risk of developing ALS later in their life. So the idea here is to understand whether there are early signals of the disease before the disease becomes symptomatic. So the hope is that we can diagnose these individuals very early and perhaps start treatment before they even get the disease. So the goal is to sort of actually in the future have the ability to perhaps prevent ALS. And another sort of important study aspect of the study is that study participation is either in clinic, so it's individuals coming to hospitals and get all the measures and also donate samples like blood or spinal fluid, but also there is a remote participant arm, so people can also be part of the study by taking assessments from their home. And in those cases, phlebotomists will visit those patients to take blood samples on a regular basis for future analysis. Next slide, please. This is a map of the all ALS enrollment sites. So the states in blue have clinics that participate in the study. There are currently 36, and one is in Puerto Rico. And the states in gray currently do not have a clinic in all ALS. But people who are interested in participating in all ALS in those states can be participants through remote participation. And we actually have very robust participation through our remote catchment. Next slide, please. So here are just some additional metrics about all ALS. I think the study really has amazing momentum. We now have over 1,600 study participants. The overall enrollment goal is, I think, 2,100. So I think all ALS is going to be fully enrolled in the very near future. And then these participants will be followed and stay in the study for at least 2 years in the prevent cohort. We hope they'll be able to stay longer. And we are really sort of happy that we have enrollment in every single state in the US. And as I mentioned earlier, we are collecting a rich collection of biospecimens. And this includes longitudinal blood samples and spinal fluid samples. And these samples are then available for molecular analyses, either commissioned by AMP-ALS, and Chubangi is going to talk about that, but also the broader research community can request access to these samples. And we are also collecting a really rich set of clinical data, and this is also a collaboration with the Critical Path Institute and the FDA. They have given us a lot of advice what types of clinical instruments should be included in the study, and that also includes patient-reported outcomes. We are also collecting environmental exposures. This, again, is a collaboration, and here the collaboration is with the ALS National Registry. This is another program that is collecting a really sort of deep set of environmental risk factors. In all ALS, it's a little bit more limited, but we really want to make sure that the data collection is harmonized, that will then in the future allow broader meta-analyses. And then finally, and importantly, we are also very much interested in digital health technology data. So all ALS is currently collecting speech data as well as movement data to advance sort of that research objective. Next slide, please. So this is sort of my last slide. I just wanted to highlight that all ALS and AMP-ALS are really synergistic research efforts. And the samples that are being collected by all ALS are going into the BioSend repository. They're already being sort of requested by the broader research community. And as we mentioned, the data is going into the knowledge portal. However, I just wanted to take a moment that all ALS is not the only sort of high quality, large natural history study that is currently active. There are also other efforts in the US. So for instance, ALS TDI has a really excellent natural history study called the ALS Research Collaborative. And I think that is through remote participation. Then also Target ALS has a really excellent natural history study. Target ALS also has international enrollment sites. And there are also then the natural history study that the FDA is supporting with Dr. Walk as the lead principal investigators. And we are really trying to sort of harmonize data collection. So in the future, all of those data can be pulled together. But that being said, I think each of these natural history study has sort of a unique strength and a unique sort of approach. But the overall goal is common across all of these natural history study, and that is to accelerate drug development for ALS. So I think all over the ALS field is really collaborative, and we're really sort of excited about bringing these research efforts together to catalyze ALS research. So this was my last slide. I just have an acknowledgement slide. So this is sort of our core team at NINDS. My colleagues Amy Zhou and Dina Lyon in our clinical division are very much managing the clinical ALS research efforts on the NIH side. And then Elio Peraza is our overall ALS program coordinator. And we also have a very nice website called Focus on ALS. The QR code is on the slide. And the community can sign up in a listserv to get updates from NIH about our ALS-related research efforts. So thank you very much, and I'll hand over to Shubhangi.
Thank you, Amelie. Next slide, please. Okay. Good morning, everyone. Thank you to the Critical Path Institute and FDA for bringing us all together. It's an absolute privilege to share a virtual stage with our colleagues from the FDA, the NIH, Critical Path, and nonprofits from the field, and most importantly, the members of our ALS lived experience community. My name is Shubhangi Lal, and I serve as the Senior Project Manager for Neuroscience at the Foundation for the National Institutes of Health. Our goal at the FNIH is to build bridges to breakthroughs. We were chartered by Congress 3 decades ago, precisely for moments like these, to connect the absolute best in public sector research with resources of the private sector. We believe in the philosophy that science has the power to cure, but no single organization can do it alone. By building public-private partnerships, we pool the massive financial resources and global intellectual capital required to tackle complex scientific problems. Next slide, please. Across all of our FNIH initiatives, we have raised over $1.59 billion in private funds, with 90 cents of every single dollar going directly into supporting our scientific programs. We have over 600 programs supported since inception and more than 120 active programs currently. The FNIH has been able to establish a track record of building highly efficient, pre-competitive public-private partnerships. Next slide, please. One of the FNIH's true flagship initiatives is the AMP program, or the Accelerating Medicines Partnership Program. As you can see on the slide, this model is built on a 11-year track record that spans 12 distinct project areas. It represents a massive collective investment of over $1 billion, utilizing 16 NIH institutes, covering 35 leading nonprofits and 36 industry partners. The disease portfolio itself is extensive. It's covering everything from Alzheimer's and type 2 diabetes to rare diseases, schizophrenia, and now ALS. Launched in 2014, the AMP model operates as a premier, pre-competitive collaboration designed to fundamentally transform how we understand disease pathways and how we can develop new treatments. Rather than working in isolated corporate or academic silos, the AMP framework forces a collective approach. We unite the unique resources of the public sector, including the NIH and the FDA, with private players such as pharmaceutical and biotech companies, alongside dedicated patient research and advocacy organizations. The AMP framework aims to identify new targets, standardize existing tools, and generate massive open access data sets for the global public good. The critical takeaway here is that AMP-ALS is not operating in vacuum. It sits on this exact blueprint. By leveraging this massive institutional scale, AMP-ALS can apply proven data standards and cross-disease insights to move the needle faster for ALS. Next slide, please. So you've seen the slide earlier, and this just shows that our presence today at highlights the powerful synergy established under the public-private partnership of the Act for ALS. As you can see from this ecosystem framework, and as you've heard from the speakers before, we are all working very closely on this initiative. There is a little bit of inherent complexity when you look at these moving parts, but each of these distinct components is deeply synergistic. You just heard from Colin and Amelie about the CPR&D efforts and the All ALS study. All ALS, as you've heard, is the engine that helps us implement the core research aims of the entire AMP-ALS program by providing the comprehensive longitudinal biospecimens and clinical data that the community needs. AMP-ALS is a public-private partnership between the NIH, FDA, and several life sciences companies and nonprofits that drives the focus on novel biomarkers and comprehensive data integration. I'll be going in more detail in the following slides on this. I also want to highlight that the people with lived experience, our PWLEs, play a central role in this collaboration, playing a key role on how these pieces interact with each other. Ultimately, all of this harmonized data converges directly into the ALS knowledge portal, creating a large unified ecosystem for ALS therapy development. Next slide, please. Now that we've looked at the high-level framework for our ecosystem, let's look at exactly how AMP-ALS is converting that structure into measurable impact. From the very get-go, the AMP-ALS program aimed to build a comprehensive strategy, not just to accelerate the development of effective treatments for people currently living with ALS, but also to protect people who might develop ALS later in life. The core vision here is to also think seriously about prevention strategies. To turn this vision into reality, our program is actively executing on 4 primary operational fronts. The first key focus is to build a centralized database to access high-value, de-identified, individual-level clinical and molecular data sets. This will track all stages of ALS progression and critically includes data from pre-symptomatic individuals as well. The second area we are working on is running comprehensive multimodal molecular analyses. All ALS provides both longitudinal biofluid samples and postmortem tissue when they become available, giving us high-resolution view of the disease mechanics over time. While our primary engine is the samples from All ALS, we've already had the unique opportunity to analyze samples from other major natural history studies as well. This really speaks to the collaborative multi-cohort nature of the work being done in the field currently. Another critical area is biomarkers. AMP-ALS is heavily invested in developing reliable biomarkers, not only to enable early diagnosis, but also for measuring progress and treatment response in clinical trials. Biomarkers play an absolutely critical role in early stage clinical trials. This is where we can capture early signals that an investigational agent is actually working or not, helping us monitor disease progression and reliably track patient responses to treatment. Finally, the clinical outcome assessments. We are developing improved patient-informed COAs to optimize clinical trial design and everyday clinical care, while establishing best practices for the tools currently under use. To optimize these measures, this entire effort is being closely led by CPAT and the FDA, who bring an immense amount of specialized expertise to ensure our frameworks are robust and ready for regulatory scrutiny. Next slide, please. As Colin mentioned earlier, the absolute heart and the true north of this entire effort is the patient community. The people with lived experience, or the PWLEs, are integral partners central to every single stage of AMP-ALS, moving with us all the way from the early design phase in 2023 to the ultimate implementation in 24 when we launched the program, and now for the community outreach. They're not passive participants. They actively help us shape and ground our research priorities, ensuring that real-world needs and patient-centered outcomes remain the program's focus. By directly involving them in discussions about our research goals and initiatives, AMP-ALS ensures that the program continuously addresses the issues most relevant to their actual needs and well-being. The real-time feedback has ensured that our clinical studies remain practical and minimize unnecessary burdens on participants. As you can see on the slide, we've had more than 20 dedicated AMP-ALS lived experience meetings since the design phase. As shown in the organizational structure at the bottom of the slide, PWLEs are fully integrated into the AMP-ALS technical working groups. They participate in these technical working groups to provide real-world feedback. Today, more than 30 PWLEs have directly helped shape the program, and their active voices also enhance several groups within all ALS, such as the recruitment and retention working group. For instance, while designing the digitized ALS pilot study within the All ALS Consortium, our PIs were able to reach out directly to the PWLEs within our dedicated working group to pull from their lived experiences in other past studies. Another fantastic example of this collaboration in action was their direct hand in providing feedback to the patient dashboard for the All ALS website, ensuring that it remains intuitive, accessible, and serving the community needs. Ultimately, our communications and program outreach are also vastly strengthened because they are guided by the very community we are here to serve. Next slide, please. Okay, the formatting has changed a bit, but that's fine. Now let's look at the actual progress and milestones the program has achieved as of mid-2026. As you just heard from Amelie and from Colin in their presentations earlier, there is an incredible amount of momentum happening on the ground with both the All ALS cohort and the COA frameworks. My goal here is to pull back and look at how these massive efforts are working to drive measurable impact across the entire program. First, on the knowledge portal and data harmonization. Following its successful launch in August 2025, the ALS Knowledge Portal is officially operating as a centralized hub for multi-omic and clinical data. We've already integrated more than 20 high-priority extent data sets alongside incoming All ALS clinical data into a single unified, highly accessible ecosystem for researchers. Second, on our All ALS natural history study, as Amelie highlighted just a few minutes ago, through a mix of in-clinic visits and remote tracking, enrollment has been incredibly rapid, reaching close to 1,600 active participants as of April 2026. Third, We have several biomarker initiatives in progress. We recently completed a high throughput proteomic study using cross-sectional samples from the Answer ALS and Target ALS cohorts to identify novel disease signatures. These studies are incredibly important because the signatures we are finding will directly inform our subsequent longitudinal studies within the active all-ALS natural history cohorts. Additionally, we have funded 2 project teams this year to develop antibodies designed to detect cryptic peptides, which are a known characteristic of ALS. This is of great interest to the field. Once developed, these antibodies will be shared openly within the community for research use. Now that longitudinal all-ALS samples are becoming available, we will be launching a series of biological analyses to ensure the samples donated by our participants are utilized to their absolute highest potential. By leveraging cutting-edge technologies, starting with proteomic studies alongside NFL measurements and whole genome sequencing, we are positioned to discover a wealth of new biology for ALS using these incredibly precious samples from the All-ALS study. Fourth, on advancing the clinical outcome assessments, this ties directly back to the statistical frameworks and respiratory measures that Colin just walked us through. So this comprehensive progress shows that the ecosystem we are building is actively delivering harmonized data, scaling patient cohorts, and preparing new biomarkers. Ultimately, all of these milestones that I've highlighted rely on one critical thing, which is true accessibility. All of this data has to get into the hands of the scientific community. And that brings us directly to the centerpiece of the program, which is the ALS Knowledge Portal. Next slide, please. It is important to emphasize that these types of data portals are absolutely central to all of the broader AMP efforts. We are intentionally aligning our infrastructure with broader AMP network, harmonizing data structures across different disease portals to ultimately enable cross-disease interoperability. When we were designing the ALS Knowledge Portal, our vision from day one was for it to be a true one-stop shop for both academic and industry researchers to access high-value, de-identified, individual-level clinical and molecular data sets. This portal is a close collaboration with the Critical Path Institute and Sage Bionetworks. To bring this to life, our partners are executing on their distinct strengths. The Critical Path Institute is driving the clinical side, bringing in pre-existing clinical data sets, harmonizing them, curating them. And Sage Bionetworks is bringing in the molecular data sets while managing the incoming newly generated all ALS data sets. A working group is currently developing a manuscript that Colin referred to earlier, a data profiling paper that will describe the portal, the current data sets it holds, and a few use case scenarios shared by our industry partners. The group aims to submit the manuscript by the end of this month. We're very excited to see the growth from launch, where we started with just 10 datasets to 25 datasets available on the portal now. To keep this ecosystem is extremely dynamic. We have new all ALS data being transferred to the ALS Knowledge Portal on a monthly basis. Looking ahead through the rest of the year, our focus is on continuous optimization. Sage Bionetworks is currently working to enhance several key features. For anyone in the meeting who wants to dive into the portal, the live link is right on your screen, or you can simply scan the QR code at the bottom right corner to go straight to the website. Next slide, please. When you look inside the portal, you can quickly see that the portal provides a true multi-omic environment. Along with omics data, it also has extensive clinical trial data, patient registries, and robust biomarker data. This multidimensional data structure allows researchers to pivot seamlessly between clinical and deep molecular signatures. As you can see by the key data contributors featured at the bottom of the slide, the ALS community has rallied behind the centralized repository. We are deeply grateful for our partners, and we fully expect this list of contributors to expand over time. Next slide, please. As we reflect on all the moving parts of this program, we have learned that it's absolutely vital to keep our entire community connected, informed of our progress, to ensure complete transparency and to keep everyone aligned. We have launched the ALS newsletter. Looking at our timeline, our first edition was released in Jan 2026, which set a fantastic foundation for the year by highlighting our inaugural milestones and our strategic direction. Our next edition is on track to be released by the end of this month. For all the community members and partners in the meeting, this newsletter is a great tool for staying connected with our operational timelines and updates. You can scan the QR code right here on the slide to read the latest edition and ensure that you're subscribed for the upcoming release. Next slide, please. So to close out today's presentation, I want to take a step back and recognize the incredible partnership that powers the ALS program. As you can see on the slide, this effort bridges the entire spectrum of stakeholders. In the public sector, we have the NIH, NINDS, along with the FDA. Our ex officio partners include Critical Path Institute and, of course, our team at the FNIH. And our private sector footprint includes leading pharmaceutical companies and biotech companies, including patient advocacy and research organizations, all sharing their resources and expertise. Bringing this diverse group together requires incredible leadership, and we are extremely grateful to our steering committee coaches who guide our strategic direction: Dan Doctoroff, founder Target ALS, Stephanie Fredette from Biogen, and Dr. Amelie Gubitz from NINDS. Thank you to all our partners and researchers for their ongoing commitment to this program. Their vision ensures that our efforts remain perfectly aligned with our ultimate goal of accelerating therapeutic breakthroughs for patients. Thank you so much.
Thank you, Shubhangi, and thank you, Amelie. I will now briefly turn it over to Katie O'Keefe for questions and answers.
Yeah, thank you, Shubhangi, and thank you, everyone, for the presentations today, and Amelie for linking both you and Shubhangi to the work that FDA and CPATH are doing. I think it's a great overview. I would like to turn it over to Amelie a little bit to speak a little bit more about the preclinical work that NIH does, as that's really their area of focus, especially on this ALS effort. So Amelie, to you.
Yeah, so at NINDS, we have a division for translational research, and my colleagues in that division oversee a range of funding opportunities specifically for preclinical therapeutic testing. And this is not specific to ALS, it's open to all neurological diseases. But I think we have sort of seen a lot of activity in the ALS space as these new sort of therapeutic concepts are developing. And some of the programs, they focus on small molecules, so drugs that you could take orally. Others are focusing on biologics. I think there's a lot of promise here. There have been a lot of advancements. And these are the type of grants that we call them milestone gated. So there are yearly assessments whether a therapeutic concept is working out or whether we sort of need to change course a little bit. So I think they're very dynamic. And I had mentioned one of the programs where we also team up the academic investigators with consultants and also contract research organizations for specific, preclinical activities. So I think there's really a breadth of programs. Of course, it changes a little bit every year. There's a lot of expertise here at NIDS. And what we really hope is that the academic investigators feel energized and approach us with our therapeutic concepts early on so that we can discuss, certain funding opportunities and, where their program would best fit, so that we then can sort of encourage them to submit, and then it does go to peer review. But there's a lot of activity and a lot of sort of different flavors of types of activities that we would support. I think I'll leave it at that.
Thank you, Amelie. And I know we've had a lot of interest and questions in the EAPs and the All ALS forward-looking statements. We'll try to answer those in chat, just because we really want to get to this last panel discussion. We are running a few minutes over, but we hope you're able to stay on a little bit past the noon hour Eastern time if you're not booked with another meeting. So I will turn it back over to our panel that's going to lead the next discussion. To you.
I am pleased to introduce our final session of the day, which will be a panel discussion on the future of rare neurodegenerative disease research. The panel will bring together leaders from across the R&D community who are advancing work in different disease areas, but with notable points of overlap, ranging from underlying biology to infrastructure needs. Pleased to welcome back Michelle Campbell to moderate the discussion. We're also joined by Dr. Fernando Riera, CEO and Chief Science Officer of the ALS Therapy Development Institute, Andrew Rosen, CEO of National Ataxia Foundation, Dr. Brian Lynn, Research Director at the Muscular Dystrophy Association, and Dr. Penny Dax, who is Chief Science Officer at Association for Frontotemporal Degeneration. Thank you all for being here. Over to you, Michelle. Oh, Michelle, you're muted.
Thanks, Penny. Good morning, everyone. As we continue our conversation from what we've heard from our prior presenters, we have a really great panel that represents a diversity of rare neurodegenerative diseases ahead of us. I also want to acknowledge that we've been getting a lot of great questions, and we are trying to do our best to actively answer those questions during this webinar. Please know that your questions are being recorded, and we will make sure they get answered. If they do not happen today, we'll make sure an answer is still given to you at some point and will be communicated to you. So I'm going to ask our panelists to reflect on what we've talked about, what has already occurred under the first implementation of the Act for ALS, and what are their thoughts for what they've heard so far. And while they say that, I ask that they first introduce themselves to our audience. So in no order, and this is just how you are appearing on my screen right now, I'm going to start with Andrew Rosen from the National Ataxia Foundation.
Hi, Michelle. Thanks for having me. I think it's been a great morning. I mean, I'll be honest, I've learned a lot about kind of some of the details of the Act for ALS. So ataxia, as you know, certainly is a rare movement disorder. I often will describe it to people that have never heard of it as a slow-moving ALS. In many ways, it shares some of the same symptoms, but just progresses much more slowly. So we're obviously we're thrilled that there were those few words after the act, after ALS and other rare neurodegenerative diseases, because ataxia certainly fits into that. So we'd obviously listen. Some of the themes that came out this morning are very relevant for our community. Would love to see sort of so for instance, that ALS science strategy. Would love to see an SCA science strategy, right? And I realize that some of this is going to come down to resources, but exploring gaps in understanding of natural history is really important. Exploring use of fit-for-purpose digital health technologies. I think in slowly progressing rare diseases that don't have clear biomarkers, which very much describes the ataxia space, I think digital health technologies can play a huge role, and so we'd love to see more work done there. And then potentially the remote administration of clinical rating scales like the ALS FRS, but in the ataxia world, that would be the SARA, I think is also an area that we would love to see. I'll stop there, but thanks for including us today.
Well, thank you for that, Andrew. I'm going to switch over again. It's in order of how you appear on my screen to Fernando to introduce yourself and just to give some reflection so far of what we've heard today.
Thanks, Michelle, and thanks everybody for being here and the opportunity for me to participate today. Recently, I've been sort of reflecting on. I'm Fernando Vieira. I'm the CEO and Chief Science Officer at ALS TDI. I've been working in ALS for more than a couple decades at this point, and one of the things I've been reflecting on in the past couple of years is how sort of my former self from 20 years ago would be so jealous of the me today who gets to be working in this field now. We wish that we were done, but obviously we aren't. And so the framework that we're working from today with the progress that's been made and the infrastructure that's been built and is being built is really, really heartening. And I think a big part of what today showed is a lot of that, where the federal government's increased involvement and investment in ALS and other neurodegenerative diseases, and the really careful thinking about a strategic plan moving forward. So I've been excited to see that. At the same time, one of the themes that has emerged, and it's been fantastic to see the questions and comments from the people with lived experience and caregivers and stakeholders in the community. There's the tension between the urgency of delivering for people living with the condition right now, while also building the infrastructure to accelerate research going into the future. And that's the tension that we all have to live in and all have to work in and continue to be in dialogue with one another. And so meetings like these today help with that. Thanks.
Thank you. Let's hear it to Brian and your perspective from a doctor patient.
Michelle, you're muted.
It's just very faint. It's like your audio just failed, Michelle.
Get a little closer. So, to Brian, you are part of an organization that covers a range of rare neurodegenerative diseases.
It's still not coming through.
Okay, we will try this. I'm not sure if it's working. It's totally, I don't know how this will work because this is a totally different audio system we're now using.
It's fixed.
So Brian, can you talk about from your perspective of being in a broader patient group level where you have a coverage of multiple rare neurodegenerative diseases, your thoughts on what you've heard today?
Yeah, so just kind of reflecting on this morning's presentations, it's so exciting to hear about all this strong infrastructure that's been built around, collecting data, but doing it in a harmonized fashion that can work across, various diseases, and also about the clinical trial frameworks and models that CPAP has helped to develop, which is also really relevant across many different diseases. And so for me, as someone who works at MDA and, supports ALS research, but also has to think about the 300 other rare neuromuscular diseases in our portfolio, many of the diseases have neurological components as well, like Friedreich's ataxia and CMT and myotonic dystrophy. I see so many synergies across all of these various diseases, but I also see, many research efforts that are being done within these individual fields without consideration of some of the programs and experiences in adjacent disease fields. And so when I look at a program like Act for ALS, I love to, see us make it easier for many of these smaller disease communities with potentially less resources that are available to them, be able to tap into some of these programs and infrastructure that has already been built here and not have to kind of recreate the wheel independently within their own fields. And I think as we continue to develop greater understanding for each of these individual disease areas, we'll really start to see significant areas of overlap and opportunities for kind of cross-disease collaboration to really push all of these fields together at once. Thanks,
Thank
Michelle.
you. Thank you, Brian. And Penny, I'm going to have you wrap up this first question. On your thoughts we've heard today, you have been a participant on the AMP-ALS side, along with Fernando, and been part of these conversations from the beginning from the organization that you represent. So what are your thoughts on what you've heard today? And maybe you want to explain a little bit about how FTD ties into this space.
Yeah, thank you, Michelle. It's an honor to be here. So FTD is a little bit of a unique situation in that we are a rare neurodegenerative disease that is distinct from ALS, but heavily overlaps with ALS. So it's estimated between 15% to 30% of people with ALS, both sporadic and genetic, also have FTD as a comorbid condition at different levels of development. And most of the people who are genetically at risk of ALS, most of the genes, particularly C9orf72, are also FTD genes. And so as I can give statistics to the best of my knowledge, but just maybe easier is a quote of one of the families we work with whose wife died of ALS or FTD with a bit of ALS. Her identical twin died of ALS with a bit of FTD, and their father died of ALS. So if we think about designing clinical trials to prevent disease for ALS, it's one of the things our nonprofit has worked very hard for, and it's why we joined AMP-ALS, is that these families deserve to truly have their disease addressed, and that disease may manifest differently depending on the person. I think that there's a lot of really exciting times, and it was wonderful to hear this morning that we're really building on a foundation of investment in rare neurodegenerative diseases that we heard a lot across both the FDA and the NIH that most of what we heard for rare neurodegenerative diseases, a lot of that we heard from today was actually not from active ALS, but was building on this foundation of investment that's really been building over time. So I think that that's exciting, and I think that some of the specific progress, like some of the FDA's work and the investment in RDCA-DAP, has been foundational. The greater incorporation of persons with lived experience at all steps of the process, I think, is a really sea change across the entire field, and it's wonderful to see that embodied in these initiatives. I think we still have a long way to go. You know, when I think about a pre-competitive investment like this, to me, the end goal, like what is our end goal? I think it should be having the tools and the data and the infrastructure and the experts so that as new companies emerge with new treatment ideas, they can rapidly and efficiently test if they work. I think it's just foundationally important. And I think we still have a ways to go to get there. And Bernie Zipprich, I think, pointed out in the chat that even within ALS, there are many, many different biological subtypes. So how do we scale that up and address not one, but all of them? I think it is embodied as well. How do we scale up across neurodegenerative diseases, whether that's the intersection of FTD and ALS, the subtypes within ALS, or all of these other rare neurodegenerative diseases? We have a lot to learn from each other. But right now we have a lot of silos of data, silos of fields, and silos of expertise.
So thank you, Penny, for that. To my panelists, I'm going to make a blending. So we pre-wrote some questions to our audience. I'm going to try to attempt to blend the second question with some questions that have come into the chat and get your thoughts on this, because I actually think they're broader than being directed per se at FDA, but I think they are much more important questions to the broader stakeholder community in rare neurodegenerative diseases. So I'm going to attempt to blend two questions into our question, and just so you are prepared, I'm going to go from Fernando to Andrew to Penny and then to Brian, so you guys are at least prepared of your order. So we know that in our rare disease community, we need to have collaboration with all stakeholders. So patients and families, number one, as well as the regulatory counterparts, and not just FDA, but rare disease is global. So all of our global aspects, health authorities, scientists, clinicians, our industry developers of medical products, right, is so critically important to science. At the same time, we are balancing the advancement and understanding of disease, or perhaps just a new discovery of disease, perhaps a new genetic marker that is actually part of that diagnosis odyssey. So how do we leverage really the broadness needs of what we're going to need for medical product development, particularly in our drug development side, biologic sides, of how do we integrate the understanding of new biology of disease and processes that is continuing to ever evolve while we're trying to determine how to balance who to include in our trials, because all of your diseases and disorders have a lot of subtypes to them, right? It may have subtypes, may have different expressions of phenotypes as well as this entire area of unmet need that we're talking a lot of stuff about the forward thinking of clinical development, but the need in some of the more translational basic science aspect work that we do know is important and can be foundational in trying to advance what is going on from a mechanistic perspective. So what are your thoughts about why we need to work together to help advance this while we're balancing the evolution of science and really this underpinnings of subtypes, and how do we balance that when we're thinking about what do we really need to know if we're accurately describing like natural history and selection in the trials?
My goodness, that is a very big question.
It's a very big question with a lot
It
of
is.
parts, but I'm
It
hoping you
is.
kind of got something out of it, and
No,
everyone
and
can highlight it parts.
it's the right question to be asking, right? So I think there are many different lenses through which we can look at these conditions and the data that we're collecting around them. And two of those lenses are: how does the disease clinically manifest, right? So what are the symptoms and what is the sort of clinical state, and how is the quality of life for the patients? And their caregivers and their whole sort of ecosystem around them. And there's another lens, which is sort of the biology, the underlying biology, be that genetics, be that transcriptomics, proteomics, and cell biology, all of that. And I think it is really important when we arrive at insights into either of those lenses in any one of these conditions, that we try to look across and ask ourselves how that insight might apply to another condition. So that is to say, if we're looking at digital outcomes using accelerometers, for example, in the context of ALS, it is good to interact with a clinician. An example of this, Anupam Gupta, who's a clinician who does ataxia work, came to us at ALS TDI to say, okay, let's analyze your actigraphy data the way I look at it for ataxia. And there was an interesting signal that emerged. On the other end of the spectrum, we know, to bounce off of what Penny described in the relationship between FTD and ALS, we know that there are certainly genetic relationships between ALS and FTD. And so if we're deriving insights in the context of ALS, we'll look directly across at FTD and connect. Those insights are likely going to apply. And it's not just ALS and FTD. There are other rare subtypes of ALS that will likely have overlap with other rare neurodegenerative diseases. So it's keeping our eyes wide open and just taking that one step further every time within that plane that we're studying to look at the other disease and make the connection, and then find the resources that might be able to do something with that. Like, an ALS group doesn't have to do all of the work for the other condition. They just need to help, position that. So that's it's a lot more complex than that, but that's where I'm at as a first blush.
Thank you. Andrew, what are your thoughts?
Yeah, building a little bit on what Fernando just said. Listen, this is a resources challenge, right? Especially for the rarer neurodegenerative diseases. So NAF has been around since 1957. We are coming up on our 70th anniversary next year. The first spinocerebellar ataxia gene was identified in 1993, so almost 35 years ago, and we continue to wait for the first treatment. It's a money and resources issue, and so we are totally dependent on this sort of ecosystem of of other rare diseases that may be better funded and maybe better understood, or at least better recognized and aware for, the world is aware of. It's so important for us to continue to rely on those. We depend on our pharmaceutical partners to help fund our natural history study. We can't raise enough philanthropic money on our own to do that. They need the natural history data, well, and tend to be better funded than patient advocacy organizations, and so we ask them to help fund it. That's just an example of why this sort of, if you will, collaboration, Michelle, is so important to us. So for an organization like ours, and by the way, we are celebrating, we just had our first, I think $10 million revenue year ever, but $10 million in this space is not enough, and we're thrilled we've gotten there, but we need to continue to grow. And so for us to kind of relying on this broader ecosystem is essential.
Thank you for that. Penny, your thoughts?
I agree wholeheartedly with what my colleagues here just shared. So I'll comment on a different piece that I think we need to maximize the learning from every single study. And that has a lot of different pieces in it. That means that when we invest from the NIH in things like natural history studies or biomarker development, we need to be incorporating regulatory science and expertise so that those folks who are doing that type of development, even if in an academic lab, have some insights into how this might be accelerated for development for industry's use. When we're thinking about if somebody joins a clinical trial that maybe an industry is sponsoring, it should not be that difficult, and it's frankly easier in the ALS space than in most places, to have placebo-armed data being shared, right? If you ask a patient to go through a placebo exposure over multiple years in a condition that is fatal and progressive, by the time they reach open label extension, their quality of life may have deteriorated to the point that they're not even going to stay in for open label extension. And they do it because they don't know if the treatment's going to work, right? We need to do the science, but we should be maximizing the learnings from every trial, and we should be figuring out how to work across all the different sectors here to maximize that learning. And the other aspect of maximizing our learnings from every individual study and trial is about these cross-sector and cross-disease efforts. So I think about the example of FTD and ALS, that we still don't know if you are a C9 ORF72 carrier, what is your likelihood of developing FTD versus ALS? Because there have been massive studies done in the FTD field and big studies done in the ALS field. But generally speaking, this data architecture is separate. And so the clinical groups tend to be separate. That is, in a world of data, right? Data is driving our economy. We should be able to solve these things with data interoperability and thinking ahead for those types of collaborations. And not just with FTD and ALS. As Fernando said very well, it's not just FTD that overlaps with ALS. There's a lot of rare neurodegenerative diseases. And I truly believe we will get faster to solutions for all of them when we have more robust collaborations.
Thank you, Penny. And Brian, what are your thoughts on the very wild and broad question I asked?
Yeah, so I don't have too much to add above what my fellow panelists have already mentioned, but just coming from like an umbrella organization perspective, a lot of the scientific advancements that we have in some of these disease areas are based off of learnings that happen in other disease areas. So for example, some of the treatments that we have in SMA have served as a framework for using some of those genetic technologies in other disease areas like ALS and some of the muscle diseases as well. And when we think about ALS and many of these neurodegenerative diseases as heterogeneous, diseases, we can take a lot of our learnings back from experiences from cancer and oncology, where basket trials are run, where a tumor appears in a variety of different organs, but the shared pathology or the shared genetic mechanism is identical. And so they run these trials that allow that drug to be tested across many different diseases. And when we look at neurodegenerative diseases from outside of kind of the diagnostic labels, we see a lot of similar molecular processes. We see repeat expansions across many of these diseases. And by applying some of these initial frameworks from oncology and drawing from other neurodegenerative diseases, we can really start to figure out a path forward and discuss regulatory innovations for some of these really small populations in terms of creating a lot of treatments for these.
Well, thank you. I hope our audience is willing to stay on for an extra 5 minutes as we wrap up this session. I know we're at time, but I do want to ask our panelists one final question. I think that is extremely informative throughout the conversation today and in the questions that we're being asked in the chat. And thank you all for your questions. You know, there's still a massive gaps and holes in the science for rare neurodegenerative diseases. We still need more preclinical work. We still need more biomarkers or the identification of biomarkers if we don't have one, if there one's available. We also are still exploring, optimizing, and appropriate utility of various outcome measures and ways to capture that data, either through a technology of a DHT or through more of our more traditional based methods of clinical outcome assessments. All of this is needed to inform not only natural history, but how to develop a trial, how to develop a study design, how to select the optimal patient population, and then ultimately, hopefully, we can be successful for a successful regulatory decision to be made. My final question to you is, from all of that, looking forward, if we are able to get the next round of Act for ALS to occur, what would be the number one idea that you would like to see to be explored in future efforts for rare neurodegenerative diseases that may be actionable and attainable? So, Penny, I'm going to start with you. I'm going to go to Brian, Andrew, and then Fernando.
Give me just a moment, please. Could you get back to us?
Okay. Well, then Andrew, I'll start over to you. Do you have thoughts of what one thing you would like to see that would be actionable and potentially attainable?
Well, listen, a quick shout out. Happy to see that advisory committee meetings were discussed at the FDA roundtable yesterday, and we think those are essential to the rare disease space and the FDA drug approval process. So, the patient voice, it's a mechanism for the patient voice to be heard. So just wanted to shout that out. I will say, and this kind of reiterates what Penny said earlier. I think patient advocacy organizations have been very generous with their natural history data sets, with both sponsors and the FDA. We need sponsors to return that favor and contribute their placebo and trial data back to kind of centralized databases. And so I think that's one area that this space would really help sort of move this space forward.
All right, great. More data sharing. I love that. Penny, have you got your thoughts or not? I'll skip you again.
Yes, I do. Thank you. To be very specific, I will say that I think that continued investment in the FDA and not just because I love you, Michelle, but I think that appropriations, the FDA's ability to fund research that's going to meet their needs for clinical trials is really important. Their ability to fund the RDC ADAP and their ability to work on things like regulatory flexibility, because in some cases, in a lot of cases, for the trial expectations for rare neurodegenerative diseases, they're not currently feasible. So how do we get past that? It needs to continue to have this deep collaboration and resources at the FDA to help us as a field overcome those challenges.
Thank you for that. Brian?
Yeah, I think, we recognize that for a lot of these diseases, there's kind of this critical therapeutic window that happens pretty early on that, once you pass this window, you can't really fix a lot of these diseases. And so I think a lot of the efforts with like the Prevent All ALS study and some of these things that are looking at like risk factors associated with these diseases should play a huge part in kind of the research moving forward. Because if we're able to move the diagnosis window just a little bit earlier, then that can have such a significant effect.
Thank you for that. I think that underpins the need for the continued development of biomarker and diagnostic criterias, as well as how do we get people earlier into being screened. Unfortunately, a lot of our rare neurodegenerative diseases, sadly, is a process of elimination, confirming it's not this one disorder and confirming on if there's not a family history per se of disease or not a genetic component that's at least been identified. So Fernando, you get the last say of what you think would be an actionable and attainable idea going forward.
Well, I'd echo everything that my co-panelists said. I think all of their points are super salient. I guess one additional thought would be to leverage the wins, right? So in ALS, we have had a recent approval of a drug that's been effective for a small subset of people with ALS, SOD1 ALS, tofersen. And I think there's a lot that could be learned, both in terms of understanding the ability to measure those benefits in the context of disease, in terms of our outcomes, and also biologically, seeing if we can, the same omics that we're applying to the context of natural history, apply those omics to the context of natural history plus effective drug, and see how that might teach us to move forward with the other subsets of the disease and other neurodegenerative diseases as well. And so I think there's just a lot of opportunity ahead of us right now.
Thank you for that, Fernando. And I do agree. I think we have a lot of opportunities going forward that we can also leverage again from those successes to hopefully accelerate some of those opportunities as well. I want to thank you, my panelists, for being here today, and everyone in the audience. I'm now going to turn it back over to Colin Huvinka to give some quick closing remarks.
I want to echo those thanks. I really appreciate everyone's time and energy today to give the update, and also just the work and input people have done for all these different initiatives. We're receiving a lot of feedback and a lot of responses, both in emails and chats and other things, so that I want to make a point that if we missed any questions, we'll be going back through these and making sure that we follow up with the individual they're most applied to try to get a response. We want to make sure that the community gets the questions that they have asked addressed and whatnot. I want to thank everyone. We're really looking forward to the further discussions about the reauthorization and the next steps and the continued progress that we're doing. And I want to thank you for your support and your time today and listening in, and your continued ongoing support. Thank you.